This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 677, recorded on October 29, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
Who else at the beginning of every Friday TWiV for some time now?
You know, I was looking back. It goes back to March of 2020.
Wow, that's a long time.
Yeah, so it's been, and it's, you know, you've been great, Vincent. You've been consistent every time. And, you know, as I was going to say, you're looking good. I think you must have discovered that Zoom feature where you click on it and it makes you look more handsome.
Certainly isn't me. But, you know, Daniel, they say consistency is the last refuge of the unimaginative. That's a quote for you.
You know, I also think consistency during these times is something that's nice. I think a lot of people have gotten into a ritual. Every Sunday morning, they go and they find our podcast. I'm going to say our TWiV.
You bet.
And they listen, you know, and I think that hopefully this is something helpful. Hopefully this gives people something that gives a little structure, reliability, predictability to what has become a very unpredictable time.
You bet. Okay, let's go.
Okay. So I guess when this drops, it'll be November 1st, right? So let me start with my quotation. To everything there is a season, a time to every purpose under the sun. So perhaps people will, some of our listeners will probably know where that comes from. But I'll leave that, you know, people can email in. But as we go forward, people will get a sense of why I'm starting off with that quotation. And I even sent out a little Twitter teaser for this episode, for my little intro. So people can, if they don't follow me on Twitter, they can track me down. And there's a little graphic that you can look at, maybe gives a little bit of context for why I picked that quotation and some of the stuff I'll be talking about today. But, you know, I'm going to start off with a patient update that actually a friend of mine, I think I'm actually going to mention him another time during this episode. He's my honorary cousin, Peter Dates. He and I sail together. We race sailboats, usually during the winter. I don't know why we don't sail much during the summer, but traditionally on Sundays when I've been less busy before the pandemic, we would race sailboats out there in the frozen waters on Sunday. So my honorary cousin, Peter Dates, was relating some stories about, was really a friend of his who went camping in the Dakotas over Labor Day weekend. Both he and his friend ended up getting infected, getting COVID-19. Unfortunately, his friend died. And so, but the other friend of my cousin actually ended up getting treated, you know, has not died. But he was curious because, you know, here we are now, and I think people who listen to TWiV have a sense of what the standard of care is becoming. But what I was shocked by was here his friend was given first hydroxychloroquine, a little bit of azithromycin thrown in there, and basically told, hey, there's really nothing else we can do for you. Good luck with that. And I think we've come quite a long way. So, you know, maybe just, you know, if you listen to TWiV, I know someone referred to themselves as the quivers of TWiV or something along those lines. But, you know, I try to do my best to disseminate what is the latest that we know, sort of my physician update just here in the first 20, 30 minutes of the show. So I'll try to keep doing that. So hopefully we can get people, I don't know, sort of up to speed with what we've learned, because we have learned a lot. And so we shouldn't be treating people now the way they were treated back in March. So I'll start off. So as we're recording this, we're recording on October 29th. It's my wife's birthday. So everyone can donate at Parasites Without Borders, you know, in her honor. But just where are we? So just pulling these numbers off the front page of the New York Times. Yesterday's numbers, we had 81,457 new cases, a 41% 14-day change. We're up 41%. And we had over 1,000 new deaths. So that was a 9% change over the last 14 days. So this is not great. For all the nurses, all the other people I work with, wear a mask. But let's move back into the schools. Now, I'm not sure if I will ever get tired of discussing schools and COVID-19 while I have school-age children. Those of us, we either have school-age children or we're affected by school-age children, or we're grandparents and our kids have school-age children. But one of my earlier quotations, and here I'm quoting myself from TWiV 635, Mask Hysteria, which was released June 5th. Apparently, it's quite a popular one. It might be the title. And my quotation at the time was, "Open the schools, not the bars." So I want to use some real life examples of how this can be done safely, and why I think even as the numbers seem to be going up, we can actually have kids in schools safely. And so I'm going to start with your alma mater, Cornell, Vincent, which I actually think is a great example of how testing can allow a university to, and I'm going to, quote was the headline of this article, or the name of the article, chalk up a rare COVID-19 containment victory. This was a nice article that was actually in Bloomberg News about this success. And we've talked a bit about Cornell, I think partly because, Vincent, you have a personal connection, but also as my honorary cousin Peter Dates, his two daughters, twin daughters, actually graduated from Cornell. So he keeps me updated. And Cornell University reopened its sprawling Ithaca, New York campus to a population of about 24,000 people in late August. This was unique, as no other of the Ivy League schools had attempted to bring back its entire student body all at once. Not every student came back. There were certainly students that opted to attend the fall virtually. But just as the doomsayers expected, people show up and a cluster of cases emerged just days before classes were to begin. But they jumped in with testing, and they've been doing testing, including pools up to 24 at a time. And I think they've really actually had a tremendous amount of success. Here we are, when this airs, we're going into November now. And they've been actually able to continue to educate our students. So it just sort of shows you what you can do at a university level, at a college level. But I just want to make a distinction that there are two real different scenarios. There's the K through 12, and there's the college university, right? And I enjoyed reading this article, because they use some of the visuals that I use, the cruise ship on land, these college campuses. But the other side of this is the K through 12. And that's actually a little bit different, right? So I want to reinforce the data, which is demonstrating repeatedly that there is a very low or negligible risk of transmission inside the schools when correct measures are put in place. I'm going to knock on wood, right? Create some sound background for you there, Vincent, when I say this. But last time I spoke about my friend Scott Burson, who's down at one of the South Shore Long Island schools, Seaford High School, and they've not had to close for a single day. And what did they do? They did a few things. One, which I thought was pretty creative. I was out walking the dogs early today in the pouring rain. The hurricane is now reaching us. And one of the things they did is they have, half of the school go to class in the morning, half of the school go to class in the afternoon, avoiding the really high risk activity of feeding and having people eat in a setting. Because as we know, when you're eating, when you're drinking, off come the masks. They've also done all the distancing. So they have the students 6 feet apart. They have them wearing masks. I'm always a little shocked when I hear about mask breaks, which is, we don't do that in the hospital, by the way. I'm not sure it should be done in schools. We keep our masks on. It keeps us safe. It keeps other people safe. They upgraded the ventilation systems. They're keeping windows open. They're probably going to spend a little bit more this winter on the heating bills. But if you do it right, when someone ends up testing positive and you ask that question, has there been an exposure? The answer for the school should be no. When you find out that someone in Johnny's class tested positive, you should say, well, Johnny was not within 6 feet for more than 15 minutes. Without a mask, there was no exposure. We should be able to continue to educate our children. We should be able to continue to do a lot of things. We have learned a lot This is just like the story I mentioned in the Dakotas. We should not be treating situations the same way now as we did in March. We have learned quite a bit. I'm hoping here in Port Washington and my local schools, they've actually gotten one of my colleagues, Dr. Wilkins, on board to help guide the process here. And hopefully they're going to stop shutting down every few days and my kids can go to school, because I love my kids, but I love my kids even more when they go to school every day.
Wonderful.
So let me go into some treatment updates. And what I want to do is, I've been talking for a while about our phases, how, a bunch of us have been using different terminology. We've been trying to get together on the same page. And so we just submitted to Annals of Internal Medicine our paper, the stages of COVID-19, 30 authors from around the world. You can imagine how much work it took to get 30 people to agree on the same terminology. Some of us had to part, with our beloved terms. Other people sort of gave and took as this went on. But what I've been noticing, and I think this is positive, we are learning a lot. And there does seem to be a growing appreciation that there are two major periods of COVID-19, a viral replication period, and then a second inflammatory period. But it gets a little more sophisticated than that. So I tweeted a little bit of this out there, so people can actually find my Twitter feed. But we tried to get together, and we actually accomplished a framework where we try to identify clinically relevant stages of COVID-19 and come up with 3 periods and 5 phases. So as I go through in future episodes, as I talk about different strategies, the when will be relative to these periods. So the periods are the pre-exposure period, the incubation period, the detectable viral replication period, and then come the phases of the disease, the viral symptom phase, the early inflammatory phase, the secondary infection phase, the hyperinflammatory phase, and the tail phase. And the early inflammatory phase, this is sort of a compilation of what some of us were calling the cytokine storm phase. Some of us were calling the pulmonary phase. This is also when the hypercoagulation begins. We're beginning to realize that even though we didn't really see the thromboembolic complications till a little bit earlier, they were actually at a small level involved in the pulmonary and the cytokine storm manifestations. So our hope is that this common terminology is not only going to serve as a framework to guide therapeutics, but it's also going to help when we do trials. Because we're going to talk a little bit about some trials that gave us some negative information, not because the therapy, I think, was wrong, but the timing was off. So let's start with the period of pre-exposure. So what am I talking about when we talk about when 30 authors agree on this pre-exposure period? This is when you have susceptible individuals, and this is your opportunity to employ a variety of measures that can minimize the likelihood of an exposure to potential infection with SARS-CoV-2. So this is thanks to people like Vincent, educating people about the virus and what we can do. And this is also something we as physicians can do on a regular basis to educate our patients. The best thing you can do is not get exposed, is during the pre-exposure period, do everything you can to reduce your risk. So what do we do during this period of time? We have masks, distancing, ventilation, cleaning, hygiene, keep washing those hands, potentially chemoprophylaxis, minimizing contact with potentially infected individuals. Testing can help to identify those, and actually optimizing management of pre-existing conditions such as diabetes, hypertension, asthma, COPD. So, in a sense, we've been falling down on that latter part. A lot of patients are afraid to visit the doctor. You might want to balance that against poorly controlled diabetes, about not controlling your hypertension, about putting on those pandemic pounds. Being in good health is a great thing to do should you be at exposure. This is also a time for active and potentially passive immunization. And by passive immunization, I'm sort of touching a little bit on those monoclonal antibodies, to which I'll return. But, one day we have visions of chemoprophylaxis where, pre-exposure prophylaxis, where maybe you're going to be taking medications before you're exposed. We see this in other diseases. But I do want to bring up what is perhaps controversial to many of our listeners. And this is about vaccines, right? This is when you want to get vaccines out there. And I've been called a vaccine pusher, and that's okay. Call me anything you want, just call me. And this was something that actually was in the news recently. So I'm gonna read a quotation from Marian Gruber. People can Google who she is. But we are concerned about the risk that use of a vaccine under an EUA will interfere with long-term assessment of safety and efficacy in ongoing trials and potentially even jeopardize product approval. Continues. And not only the first vaccine, but maybe even follow-up vaccines. So there's a lot here. And I'm sure people will, when they start to think about this, they'll start to realize what is being talked about here is delaying EUA of vaccines. We sort of gone back and forth on this, right? First, there was a rush. We're going to get them out there, as soon as someone, comes up with an idea, to even when we start getting efficacy and preliminary safety data, realizing that if we release those, that's going to be an issue moving forward, looking at other vaccines. So just to throw that out there, all 4 of the major vaccine trials are, ready to be fired back up. So we actually are anticipating interim results to follow in November, December. Novavax actually delayed the start of their protein-based nanoparticle vaccine phase 3 trial. They're having production issues. Flu vaccines, we're going to vaccine push here, get your flu shots. There are some regional short supplies, but it looks like nationally we're okay. So it's sort of a distribution issue here that we're trying to work through. But having that flu shot, having less flu circulating around is going to be a huge boon for us this year. Let's move into the incubation period. Now, this is kind of a tough window because once you've had the exposure of being within 6 feet for 15 minutes, and that's cumulative, right? You can't just step away and reset the clock. You may or may not be in this period, right? You sort of don't know if you were in the incubation period until afterwards. So you have this presumed incubation period when, perhaps you're quarantining because, at any point during the 2 to 14 days, you might get sick. So, we're hoping that actually this is a period of time when we can start jumping in with therapeutics. And when I get to the monoclonals here in a minute, this is actually something in high-risk individuals we might consider looking at. So there are some discussions about trials there. But then you move into a little bit more understandable phase, the period of detectable viral replication, right? And this is followed soon thereafter by the viral symptom phase. And this is really our chance to keep people out of the hospital. And here I'm going to bring up the Solidarity trial because a lot of people found issue with this. They went through and basically it was a study where they said, nothing really works. And included in there was remdesivir. So when you look closely at the remdesivir, for instance, a lot of people are a little concerned that, they're giving remdesivir to people who are in the ICU. They're on ventilators. And so they actually broke down their data a little bit into people who are on ventilators, not on ventilators. And you could see a lot of the data, if you give remdesivir to someone already on a ventilator, it's not helpful. If anything, you're better off not giving them remdesivir. Toxicity, liver toxicity, no clear benefit. So, one of the things is you hate to spend, and this was millions and millions of dollars, not just 1 or 2. Millions and millions. You hate to do that and see them look at a drug at the wrong time and then basically come away with, it doesn't work. This period here, the period of detectable viral replication, the viral symptom phase, is also the time when we think that the monoclonal antibodies might play a role. So people have probably noticed there was a little bit in the news about this lately where, again, they said, we looked at giving monoclonal antibodies to people who are already in the ICU on ventilators and we stopped that trial. That's a good thing. Doesn't mean monoclonals don't work. It just means really bad idea to give an antiviral therapeutic after the virus is mostly gone. Right. So I'm sort of glad that they stopped that, but I hope the takeaway from that is, again, not sort of the solidarity nothing works. We're back in March. So we started actually talking about monoclonals back on TWiV 611 back in May. This was Corona and Crown. And since then, we've had a little more developments on this front. So we had talked back then about a conversation I had had with the people at Regeneron. They had started a trial back then where they were giving monoclonal antibodies to what stage of disease? The inflammatory stage, people in the ICU on ventilators. And at that time we said, this is a terrible idea. They said, but we don't know how to do those other trials. We don't know how to do trials in the outpatient setting. We said, all right, let's make this happen. So fortunately, those trials have been ongoing and now we're seeing the fruits. So let's talk a little bit. What are antibodies? This is a little bit tricky, but that's okay. This is a very complex protein that is made by your B cells once they differentiate. I always think of Richard Nixon. You know, he's got the two fingers up and he's shaking his jowls. And he's basically trying to educate the American public about antibodies. Good job, Vincent.
He used to do double. He was bivalent.
Yeah. And actually the nice thing is a lot of these companies are studying the right hand shaking one and the left hand at the same time. So Eli Lilly has a couple of these going on. Some of them are coming from human. Some of them are coming from the $120 million humanized mouse. We've got Regeneron, right? They're studying their combination of two monoclonal antibodies. You know, the quote unquote, the Regeneron that our president received. And we actually have a Vir GSK is working together. And they've actually got one where they've modified the hand part, right, of the stalk part, where it gives it a slightly longer half-life. So once you give someone the therapy, it may continue to protect them a little bit longer. The data, I have to say, is really pretty compelling. And a couple of these companies are going to the EUA. If you give these individuals, we've talked a little bit about this, but if you give these to high-risk individuals, someone over the age of 65 with even a single comorbidity, we're looking at a greater than 70% reduction in this person ever needing any medical assistance, ever developing hypoxemia, ever going to an ER or hospital. So they're going to price them accordingly. Because that's pretty huge if we can take our high-risk population and the majority of them never end up in the hospital. I look at Vincent because I'm wondering if he's thinking about how does this work? Well, I'm involved actually, full disclosure, helping Eli Lilly do really a sort of a post-marketing type study if the EUA goes through. And what we're going to do is we're going to use Optum Infusion. If someone has a positive test, we'll send the nurse right to the person's house. They'll put a little IV, they'll get the medicine, that'll be it. So we're going to be using couriers to help improve access in the older adults. I've been told not to use the word elderly, but improve the ability of older adults to get testing right in their home and actually have these therapies, quote unquote, delivered to the doorstep. Because these are the individuals that are highest risk. And if we can keep these individuals from ever needing to go to the hospital, this will be tremendous. So very encouraging stuff here. I'm going to jump ahead to convalescent plasma. I'm going to try to keep this short today, right? I think I mentioned everyone, this is my wife's birthday. So convalescent plasma. This was another, we'll say, failed trial published in the BMJ, convalescent plasma in the management of moderate COVID-19 in adults in India. It was an open label, multi-center randomized controlled trial, cute name, the PLACID trial. 464 adults over the age of 18 admitted to the hospital with confirmed moderate COVID-19. 235 were assigned to the convalescent plasma, and then sort of compared to standard of care. And they didn't find any benefit in this study. But a couple things I'll say. The role of plasma, remains uncertain. But in this study, 79% of the patients enrolled already had detectable antibodies. They're giving this to people after the stage when we think it's going to make a difference. So, again, I'm going to repeat, these are big, expensive studies. And I'm going to argue, as many of my co-authors do in the paper we have out, that we're spending huge amounts of money on trials that are doomed to fail and provide misleading results if we're looking at the wrong phase of the disease when we do our trials. So let's move into the early inflammatory phase. This seemed to be the tocilizumab week. There was a flood of papers. I don't know if people are following. And I sort of took my notes on this section and I redid it about 4 times because there were so many papers that were sort of flooding through. But I'm going to pick one paper to discuss, which was actually our experience at the Northwell hospitals. And this is where we used a combination of steroids, where we would give steroids and then give the tocilizumab. So I think our regular listeners perhaps recall that this has been my bias of, a background of steroids first, and then you add toci rather than giving toci without this background of steroids. Tocilizumab is an IL-6 receptor inhibitor. So we're trying to interfere here with the cytokine storm during the early inflammatory phase. And so in this paper, which is actually out on a preprint right now, comparative survival analysis of immunomodulatory therapy for COVID-19, cytokine storm, a retrospective observational cohort study. So we see good and bad things in the title. I love the cytokine storm terminology. You notice it's not really in our phases. It's sort of hidden in there. And I really love the cytokine storm terminology. But the nice thing here is this study looked at over 3,000 patients, and they looked at people giving steroids and then tocilizumab. So what happens when you did it in that context in this experience? So the patients treated with a combination of tocilizumab and corticosteroids, and it was steroids followed by tocilizumab. Their hazard ratio for basically dying dropped down to 0.46. So they had improved hospital survival compared to standard of care. This translates into a 54% mortality reduction over standard of care. Steroids alone gave you a hazard ratio of 0.7, so about a 30% reduction in mortality, which was consistent with other studies. So steroids drop your mortality by 30%. And then when you add tocilizumab afterwards, the mortality reduction was 54%. So I thought this was pretty impressive. And there is a really nice, if we go to figure 2, there's a nice Kaplan-Meier plot in this paper. So you can see different approaches and how they did. Anakinra, that was an IL-1 inhibitor that was being used in a lot of the hospitals in the Northwell system. If you got monotherapy with anakinra, your survival was about 30%. If you got steroids alone, your survival went up to 45%. And if you got steroids followed by toci, the survival was 60%. So this was data out to 30 days. But a couple, let's caveats here, because I don't want, people to think like, I'm a cheerleader for tocilizumab. This was retrospective. It's observational. And we do get to see this combination of steroids and toci, but now we need to see steroids, toci, randomized controlled trial. And there are at least 5 additional randomized controlled trials evaluating tocilizumab in patients with COVID-19 currently underway. And the interesting thing is going to see, a study where this approach, because I think tocilizumab all by itself, we're seeing a lot of data saying that's not a great idea. All right. Steroids still being widely used. And I think here we had, another bit of data that was along the lines of that making a difference. We're routinely using prophylactic dose anticoagulation, and then escalating this to full dose if we feel like the risk benefit warrants that. And in the US, we tend to use low molecular weight heparin. In places with more limited resource, it's an injectable heparin. We're also using a couple of these direct oral anticoagulants. Now there was actually an interesting article about aspirin. So certainly a lot of contexts where people, don't want to do this and they decide they want to use an alternative. So this was actually an article, aspirin use is associated with decreased mechanical ventilation, ICU admission, and in-hospital mortality in hospitalized patients with COVID-19. Title basically says it all. So in this study, only about 25% of the patients who got aspirin, who didn't get aspirin, were receiving heparin. So this is in most cases looking at people that did not get an alternative. And so this was a retrospective observational cohort study, so still not at the level that ideally we'll like, but just a little bit of support for people that are using aspirin, suggesting that, it's better to use something than nothing during this coagulation phase, which is starting during the early inflammatory. Then after the early inflammatory, we move into the secondary infection phase. This is really sort of straightforward. This is that high risk for pneumonia, bacteremia, bacteria in the blood. We have learned that Staph aureus is a particularly common pathogen during this period of time. People usually don't need antibiotics when they walk in the door. Only about 10% of people have a bacterial process with the virus. But this is a time when we start seeing the bacteremias and pneumonias, the time when antibiotics might play a role. The hyperinflammatory phase, this was originally the multi-inflammatory syndrome in children. There's now a multi-system inflammatory syndrome in adults as well. So MIS-A. And the hope we have is that earlier innovations will prevent progression to this phase. And there's sort of an overlap here with the study I'm going to talk about, the tail phase. Growing appreciation of the tail phase. But there was an interesting article looking at auto-antibodies that seemed to hit all the major news media, where basically they're finding broadly targeted autoreactivity is common in severe SARS-CoV-2. And what did they find? What did they find? So they found consistent tolerance breakdown. Normally, people's immune system tolerates themselves. That's better than a lot of people, right? I'm not sure how well everyone tolerates us, but our immune system seems to tolerate us, Vincent. And what they looked at was something that maybe our listeners are familiar with, something called ANA, anti-nuclear antibodies. And what are these? These are basically antibodies against the nucleus. So anti-nucleus, you could almost say, antibodies. And this is something you normally shouldn't be doing. You normally shouldn't be making antibodies that target your nucleus. And they basically were finding that a high percent of people not only had these, but a significant number had them at high titer. They also found reactivity against rheumatoid factor, phospholipids, prothrombin, the ANCAs. And when they looked at the different types of ANA, there's different patterns. They found what they said was predominantly a speckled pattern, about 50% of people. What does that mean? Homogeneous is when you do the whole nucleus. Speckled is you see it going after just certain areas, but not the whole nucleus. So these are what we say as targeting extractable nuclear antigens. But just to make it simple, it's a pattern we see in lupus and rheumatoid arthritis. There's nucleolar and centromere as well. But I'm going to leave it here just to say it looks like we're starting to see a little bit more interesting information about maybe the mechanism behind the tail phase and a lot of the arthritis and the fatigue and all the other symptoms that we're seeing. So it'll be nice too, perhaps, if this will give us a little ability to do some objective testing, because I know a lot of these people are suffering and being not taken seriously. And a lot of times when you're trying to work with them as far as their workplace and unemployment and issues like that, it's nice to be able to have some objective measures. So that allows us to do a little bit more objective testing there. So I'm gonna finish there for my part. And I guess I'll ask you, Vincent, if there are any emails that we need or any other topics we should talk about.
Well, there are, but maybe in the interest of your wife's birthday, we could skip the emails this time. Does that work?
We could do a couple if they're nice.
If they're nice. All right. I wanted to bring up 2 points quickly first. First, did you hear that in Russia they have a masking mandate now?
I heard that Vladimir is the first international leader to have a national mandate for citizens to wear masks.
Yes.
Kind of crazy, right?
Yeah, so there's a lot of talk about this, and it's, the Biden campaign is saying that they would want that, but they recognize there would be legal challenges to it in the U.S., so probably is not going to happen.
We live in a country that really favors our freedoms.
Yep, for sure. The other thing, Daniel, let me share my testing results with you. So Columbia does random tests of people who come here to campus.
Okay.
So last week I was selected to come, and on Thursday, a week ago, I went. They have a lovely system set up here in the atrium in front of Alumni Auditorium.
Okay.
You go in, they give you a tube with your name on it. You sit down on a desk, and there's a procedure where you swab yourself and put the swab in the tube and so forth. And then it gets sent to the Broad Institute for PCR. and they are doing a 1 primer pair PCR to the nucleoprotein gene, okay? So Friday night, I get a phone call from the person directing this program. And I'm having dinner. My wife said, it's for you. I said, I don't talk to people on Friday night. She said, I think you better talk to this. So I'm positive.
Oh, my gosh.
So what would be the first question you ask, Daniel?
So whenever we do a test like that, I always want to know like the pre-test probability. So is there any reason why you should be positive, Vincent? Is there anything you've been doing that puts you at risk? It's
possible, but what I asked her was the CT value.
I like that.
And she told me it was 34.
Can I guess that? I was going to guess it's in the 30s.
34.7.
I'm hoping this is negative, Vincent.
34.7. Okay, so Friday, she said, you need to isolate. So I said, fine. I went in my office in the basement for most of the weekend. I didn't eat with my family. My wife went to a different bedroom. So everybody's wearing face masks. And I thought it was a false positive.
That's my highest suspicion, Vincent. And we've talked about this on the show. I would consider that in someone like you as a screening test to be a false positive and would, do a repeat test.
Right.
Did they offer you that? Did they offer confirmatory testing?
So she said, come back. I went back on Tuesday. Yep. I went in, did it, went home immediately. And then yesterday, Wednesday, it takes about 24 hours to get the results, or 36 hours. And she said it's negative. So that's the story. But it just goes to show that, you get a high CT value, you really need to repeat it, right?
You really, any time you do a test where the positive predictive value is less than 50% requires confirmatory testing. For you, you could sort of, do what you needed to do. But for a lot of people, could you imagine if this was, your 11-year-old son? You have to wear a mask. You have to stay in your own room. I think, I think any, a couple issues with that story, right, Vincent? Why aren't we doing it at Columbia? Why are we sending it to Boston?
Oh, really? Truly. I agree. Yes.
And the second is when someone's positive, you want to have rapid, confirmatory testing to sort that out. So.
Yeah, no, I agree. I think you asked me, did I have any risk? Now, no, I'm mostly home and I come here to my office 2 or 3 days a week. I don't do anything. So there would be no reason. Now there's just one possibility which they brought up. Now they haven't found many positives on these random screens.
Yeah.
But the ones they have been in labs with plasmids encoding SARS-CoV-2 genes.
Oh, that's interesting.
And that's what they think is going on.
Yeah.
Now
that's an interesting twist, right? In a in this kind of a testing scenario.
Yes, all the positives have been in labs with plasmids, and now my colleague Amy Rosenfeld, who thinks that's nonsense because she said she hasn't had the plasmid around for months, but these things can linger, I think.
That's interesting. No, it's really, really interesting.
But it's interesting for labs working on plasmids. You have to be careful if you're doing random testing.
Yeah. The other thing, which is interesting, I asked you that, of course, put you on the spot on, sort of live podcast, live YouTube, whatever you want to call it, pre-recorded, right? You could always edit this out. But no, a lot of times when someone has a positive test and it was quote unquote screening, and I learned this from the days of sexually transmitted infections, you say, hey, it was positive. So do you want to share a little bit more than you shared with me before? Yeah. And then people are like, yeah, and then there's the UG and you find out. And, I think this has been actually, I don't know how they get this data because I understand how I get the data as a physician. There's a trust, there's a confidentiality. But a lot of people say that they're wearing masks. They say they're doing all the right things. And then, push comes to shove. I'm like, yeah, I'm just getting tired of washing my hands. And, me and a bunch of the guys did whatever. So, yeah.
But I have to say, I was good. I isolated, right? And did all the right things, even though I didn't think it was anything. And what are the possibilities? So false positive, but it could also be I'm at the very beginning of an infection, right before we get much RNA amplification.
Or the tail.
At the very end, and maybe I had an asymptomatic infection, right? So it could have been either. That's why a rapid second test would be good. I should have come the next day, I think, but...
Yeah, no, that would have been more helpful. I mean, the other thing you can still do is, you could do an antibody test kind of now and in 2 weeks, and that gives you a sense. If it was at the tail, the antibodies should start to come up by now. If this was, at the beginning, then your antibodies would be negative. So sometimes it's actually helpful diagnostically to have that confirmatory PCR and a serology test to help sort out what might be going on.
Anyway, I thought that would be fun to talk about.
Yeah, I'm glad you're okay. I worry about you, Vincent.
Oh, yeah, I'm in a risk group. I'm okay. But I have blood group O.
Yes. I'm A+, so I'm just I'm a god.
All right, well, let's do one email because this is a cool one about testing. This is from Will, who writes, I am a captain paramedic for a large city north of Detroit. Kathy's meteorological report is the same as mine. I'm in charge of Macomb County's Hazardous Materials Response Team. I teach paramedic curriculum to new students and CME to paramedics. I began listening to TWiV after my son had a crappy encounter with a little parasite by the name of Cryptosporidium 5 years ago. I quickly became a fan of Microbe TV and have been using info from your podcasts to aid in my teaching ever since. I'm concerned with a second wave and how the emergency response community will handle it. As with most HCPs, we were dealing with PPE shortages in the beginning. Now that grant money has alleviated this issue, we've turned our focus to building a reliable testing regimen and preventing an outbreak within our department. I plan on utilizing UHG's testing simulator to help accomplish this. The access to testing sites and the type of tests they use have been very unreliable and inconsistent. Here's a breakdown of testing results for our 100 fighter paramedics. PCR or antigen positive since March 12, antibody positive since March 9. Not all 12 went for the antibody test. We have been using PCR for guidance on when someone can return to work. The New England Journal paper on rethinking test sensitivity made me reconsider that. We have one firefighter who was PCR positive with mild symptoms last week in August. He went for PCR and serology tests last week. Both of those were positive. He's currently asymptomatic. Here are my questions for Dr. Griffin. Is his current PCR positive test likely showing post-infectious shedding of RNA from the last week in August rather than a new infection, possibly a false positive? Want to take these one by one?
Yeah, let's take them one by one. So these are helpful because I think, these are questions that a lot of people have. So I appreciate your story because you used isolation and quarantine properly, Vincent, or at least you used the word isolation. So when someone who does not have severe disease gets infected, we recommend from the onset of symptoms or from the time of a first positive test, because not everyone's symptomatic, that they isolate for 10 days. And then at the end of 10 days, if they're symptom-free for 24 hours, you're really done. And we now have millions of experiences that support that. The second is if someone has more severe disease, they end up in the hospital, then it's 20 days from either that first test or from the onset of symptoms. We no longer recommend in most cases that you do a test-based return to work paradigm. We really don't think you need to do that. This works properly. So for instance, in this individual, I'm going to say, I don't think you need to do the PCR test. We require 2 negative PCRs for entrance to a nursing home. And actually our nursing home study, I think, is going to soon be published in Emerging Infectious Disease. We used a really strict, you had to be negative PCR for the NHL experience. But in general, this is, I think, excessive. So, I wouldn't continue to have this person get tested. Repeat infections so far in the time span we have are very rare. We think we're starting to see more here in New York as the incidence goes up, but we'll see. But, within 90 days, we don't recommend that someone continue to be tested.
Okay. So his next question is, should he get an antigen test because the city will not pay for 2 negative PCRs 24 hours apart? But
I don't think you need to do this. I think you can stop the testing and move forward.
Okay. And he asked, can you provide resources for obtaining rapid testing equipment and supplies?
Yeah, so the entire Optum network is basically moving to try to create access places for everyone to do all the testing. Optum Care, right? It's a whole network, all the Optum urgent cares. So if you're having issues, what we've even done this over time is partner. You don't have to reinvent the wheel. So, when we were having this ramp up, right, we're doing 150,000 tests a day in New York, and ProHealth is doing the bulk of those. We can't do all those ourselves. So I think I've mentioned we have a line of airplanes at Teterboro, and we fill those airplanes with samples, and then Quest runs them for us in North Carolina. So, reach out, partner with people. There's ways to get this done. Don't feel like you've got to try to shoulder all this. That would be my advice.
All right. He's said he plans to move to a higher frequency testing model, utilizing rapid tests to minimize false positives and unnecessary quarantine of our members. And he thanks us for being a reliable resource. And he signs off, your friend in science, Will. That's great.
No, I think that is great. And the one thing I'll just make sure I throw in there, if you're going to be doing a lot of testing, have confirmatory testing. And we have in our calculator, and we show if you don't have the ability to do a confirmatory test, you've got lots of people that end up sitting in quarantine. So I'm going to sort of say, or isolation, right?
Isolation,
because they've been testing positive. So,
all right, that's our weekly COVID-19 clinical report from Dr. Daniel Griffin. Thanks again, Daniel.
Yeah, I'm going to thank you. But before I go, let's not leave money on the table. I'm going to say, we're right here in the middle, right? It'll be November 1st, the middle of our Microbe TV fundraising. So go to Parasites Without Borders. Your tax-deductible donation, we're going to double it up to a total donation, which I think we're going to hit $40,000 to go to Microbe TV to help Vincent and the team do what they're doing.
Wonderful.
So Parasites Without Borders.
Thank you, Daniel. Joining me today from Fort Lee, New Jersey, Dixon Despomier.
Hello there, Vincent, and everybody else. It's a little cloudy. It's just stopped raining. It's about 48 degrees Fahrenheit. And it was a soggy 3-day period just
Very
now.
soggy. We were supposed to get a freeze tonight, Dixon.
We deserve this. We should have been getting this a while back because we were in a drought.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's 43 Fahrenheit, 6 Celsius here with the same gray, rainy, about to have a frost weather.
We get the city heat bubble over in Fort Lee, so that's why we're 3 degrees hotter.
From Austin, Texas, Rich Condit.
Hi, everybody. You guys are seeing what was here.
Right.
It has passed us, and it is now 65 degrees and sunny, headed for 69, and looking out at the 2-week forecast, if you want to call it that. I mean, you can't do that, but at any rate, I don't see anything over 80 degrees, so nice. I'm feeling secure in fall.
Have the cottonwoods turned yellow yet, Rich?
I would have to go outside to find that out.
This is all true.
I can't tell you.
Okay.
Don't know.
Okay. That's pretty timely.
Nothing much is turning yet.
Pretty timely in Texas.
From what I have seen.
Yeah.
And from Western Massachusetts, Alan Dove.
Good to be here. It is 33 Fahrenheit, 1 Celsius, and we have a light snow falling.
Ooh, you really?
Yep. In fact, it's been mixed rain and snow and then light snow. I was earlier in the week, I was a little bit terrified of this storm system because they were forecasting a lot of snow.
Right.
And we had an experience in 2011 where we got a lot of snow right around Halloween. And if you've ever wondered why deciduous trees lose their leaves in the winter, I'll tell you, because when a lot of snow sticks to them, the branches all come off. We lost power for 9 days.
9 days.
So,
it ended up just mostly being rain, but it is currently snowing. We got a little accumulation on the ground.
Yeah.
Supposed to be snowing in Poughkeepsie, I think. I saw that.
No kidding.
As well, up the Hudson River.
Yes, my sister in Boston sent me pictures of snow this morning.
Of snow,
Okay.
yeah.
Goodness.
October's almost over, right? We got one more day.
This is Halloween.
Tomorrow is Halloween.
Tomorrow
Tomorrow
is Halloween.
is Halloween.
Yeah.
That's right. Tomorrow is Halloween. That's right.
All right. Well, for the rest of October,
Does anybody have all of your towns banned trick or treating?
Yeah, ours is.
I believe
Not
so.
ours.
Oh,
This is Texas, dude.
oh, Texas, of course.
So, but
You're allowed to hand out ammo now, I guess.
That's the trick.
First of all, my wife and I are shuttering the place up and probably going over and hiding deep in the bowels of my daughter's house with a couple of pizzas. And the neighborhood is putting out treats on tables in their driveways.
Okay.
Okay. And with presumably a supervised pickup, because we've tried this before, leaving a bowl of candy out on the shelf out on the porch when we were going to be gone. And it doesn't take long for somebody to come and just empty the whole bowl into their bag.
Yeah, exactly.
Nice. Well, no one comes to our house. I'm not worried about anything, but I think our town has banned it. Yeah. Yep.
I'm assuming there's somebody with a really sick sense of humor who's going to toilet paper houses this Halloween. And that's just,
Don't even suggest it. Well,
it's going to happen. You're going to see photos.
My wife came back from Costco today, well prepared for the next wave. Okay. So when you guys run out, I can
did
tell
she
you
wipe
something.
them out?
Alan.
Sorry, I'm on a roll.
Dear.
I was going to say something, but I forgot. Oh, well.
That's going to become more and more common with this crew. We're going to be relying on Brianne to carry us through.
Or
I'm going to
field
make sure to
notes.
start taking notes.
What were we just talking
Brianne,
about?
yeah.
That's right. That's right. Let's see. Which page was that on again?
I wanted to say something, but I forgot. It doesn't matter, really. If it's so transient, it's important it'll come back. But what I want to say is that for the rest of October, November, if you donate to Parasites Without Borders, they will match your donation and give it to Microbe TV. So you have another month for that. Parasiteswithoutborders.com. I also want to remind you of the new Instagram account that's been being curated by Lauren, microbe.tv on Instagram, and great summaries of all the pods, not just TWiV, but all the other ones, and getting people involved and so forth. And today we're going to give away, we're going to start a contest to give away a copy of Principles of Virology. You got to stay tuned to find out when I say what you have to do. I'm not going to tell you now. Got to listen. And if you fast forward, I'll know it. So don't do it. And I'm going to I'll do one on Instagram and maybe some other platforms just to try them out, So keep listening. Anyway, there's good stuff in this pod. You should listen anyway because you're walking your dog, right? What else
Or
are you
you're
going to do?
working in the cell culture hood or the BSL-4. Or,
I've never understood that. There's no way I could listen to this and even do something like a plaque essay. No way.
I hear you.
I can't either. But, it's funny. I can listen to a podcast in the car, no problem. But I can't listen to spoken word books. I can't focus on them. I don't know why. Yeah.
Oh.
But a pod is fine. A conversation is no problem. But yeah, you're right, Rich. I can't do it either in the hood or anything.
When I'm doing something like a plaque essay, or when I used to do plaque essays, I need to give it my full concentration because it's so easy to screw up. You try and make them idiot proof, but it's really easy to screw up.
I was once told that I should never strive to make anything in my courses idiot-proof because idiots are so darn clever.
That's good. I like that. I very much like that.
I can do it, and as long as I'm only labeling one tube. I always screwed it up.
I have a couple of news items which I just added. I'm going to call this sit rep because we used to do sit reps for Ebola outbreak. This is kind of a neat word, right? Situation reports.
Yeah.
Yesterday, there were 90,000 new cases in the U.S. That's more than one per second. It's amazing.
That's incredible.
So we've been now past 9 million total cases in the U.S. and 1,000 deaths yesterday. And curiously, on Tuesday, The White House science officer said one of the administration's major achievements was ending the COVID-19 pandemic.
Vincent, that's not the science officer. It's the seance officer, okay? He doesn't have a science officer. He has a seance officer.
And, the White House is always saying that it's just we're rounding the corner. Now, I am not doing politics
and getting
here.
hit by a bus.
This is not
See what's
politics.
happening around that corner, though.
This is not politics.
No, yeah.
This is not politics. This is science. That's wrong. We're not rounding the corner.
No, we're not.
The pandemic has not ended, so this administration can't claim it. So I don't get it. I'm a scientist.
I have read this document carefully and various bits surrounding it. And I would say that quote comes from an ambiguous point in the document. It's the heading of highlights of the administration. Okay. And it could be interpreted as ending the COVID-19 pandemic could be interpreted as a goal.
Okay.
Okay. Not necessarily an accomplishment, but I have to say it's ambiguous, but sufficiently ambiguous so that those who are so inclined can very reasonably spin it as a claim rather than a goal.
Sure.
So, Rich, my take on it is that it's a typo for that. So we're not rounding the corner. They just left an O out.
Right. Yes.
Rounding the corner.
That's right.
Either way,
That would
I
have
just
been an accurate
wanted,
portrayal.
look, in contrast to the 90,000 cases and 1,000 deaths, it's not over. And in fact, it's probably going to get worse. I suspect we're going to hit over 100,000 cases a day.
It is getting worse.
Yeah.
We're seeing, every place is seeing, well, in some cases, it's just continuation of the first wave. But for places that actually got this under some modicum of control for a while over the summer, we are now seeing a second wave. And I'm sitting here in Massachusetts where we just broke 1,000 cases a day for the past few days. And that's not a milestone we want. And it's been big news in the Massachusetts, the mass news outlets are nobody's happy about this. And we all see this as a major second wave going on. And a lot of that stuff is going to have to kick in that we talked about last time about, you reach these case counts, you've got to close more stuff down.
Yeah.
Right.
It's not going to go away until everybody wants it to go away. It has to be everybody wants it.
Well,
to
it
go
has to
away.
be. We need a vaccine.
Right. And realistically, even if tomorrow we put into place all sorts of restrictions and everyone changed their behavior tomorrow, we wouldn't see an effect of that for a couple more weeks. And so right now, the case numbers are probably going to go up for a couple more weeks.
Yeah,
Sure.
Plus however long it takes us to get our behavior changed.
That's right.
As we were saying back in March, it's a lagging indicator when you're looking at case counts. And even now, when the testing is finally up to reasonable speed and people can go get tested, it's very true that it's going to take a couple of weeks before you know what's really going on.
Absolutely. A good friend of mine lives in Melbourne, and about 2 months ago, he was chiding us for this rise in cases, and all of a sudden, they got the same thing happening to them. But they've beaten it down because everybody cooperated.
Yeah,
and that's what it's going to take. And when you've got too many,
I have virtually no confidence that we're going to get the level of cooperation required out of our population.
Yeah, I
have no confidence we're going to get the level of cooperation required from our governors nationwide, and certainly not from our federal government.
I mean, even if under an administration change we get a completely different message from the top, I think it's become the sort of divisive and politicized nature of this has become so entrenched that I don't see it. I don't see it turning around.
I mean, look what's happening in Spain, right? I mean, they're having riots because the government decided to close everything down because that's the right thing to do, and they don't want to do the right thing. They want to live their lives normally, in quotes. So I agree with you, Rich. And it's not just our population; it's the whole goddamn world.
I agree, Dixon, and that's a good point. It's people don't want to deal with this.
It's a long-term thing that no one wants.
And it's when it's only a small fraction of the population, significant though it is, small enough so that most people don't really experience the extremes of the downside. They're not working in the hospitals. They're not seeing corpses pile up. It's what they're seeing is their businesses go down.
Yeah.
For me, instead of an antiviral vaccine, I would much prefer to see an antiviral drug that you could take the moment you discovered that you were sick from this thing. And you could get over it by simply taking this drug, like you can for a lot of other things. But do we have any of those for any virus infections, Vincent?
Mm-hmm. Yeah, Tamiflu for influenza, for example.
HIV.
And HIV.
Mm-hmm.
Well, HIV is different because you have plenty of time to diagnose and treat.
Yeah,
understood.
But these acute infections are tricky because, as we'll see in a moment, boom,
brief window.
Very brief window of treatment. And
there is work on this. There people are doing that, but again, as we'll see in a moment.
It's early on. So the other bit of news I just got from the Wall Street Journal via Amy, probably it's in other places by now, but Regeneron has suspended testing of its monoclonal antibody in the sickest patients because of a safety concern. And we don't know what this is, but the Data Safety Monitoring Board saw a potential safety signal and unfavorable risk-benefit profile. So they recommended pausing. So these are the sickest patients. The other patients who are less sick are getting the antibody. That's not been suspended. However, I don't know why.
Two separate trials, right?
Two separate trials. I don't know why you are giving a monoclonal to people who are very sick and well advanced past the viral phase.
Desperation.
Exactly right.
Desperation. It's just they want to do something, and they're testing it in the people who have less severe disease and trying to get them earlier, which is where we might expect this approach to work. And then, well, you're giving it to those patients. Why aren't you giving it to these patients who are sicker? That's
correct.
Okay, fine. We'll do a trial in the patients who are sicker. And we know from Daniel diagramming the disease and discussing this whole course. If you get it early, then you're targeting the virus. But if you're dealing with somebody late in the course of disease,
that's the kitchen sink. That is the kitchen sink approach.
And Daniel said he lamented all these trials where we're spending many millions of dollars and treating at times where it's not going to work. We're using an antiviral approach. So he prepared this graph, which we'll put in the show notes, which shows the course of an infection starting with early days, incubation period, the rise of viral titers, and then the symptomatic phase, and then rapid decline of virus titers. And then you enter this inflammatory phase where the viral titers are just dropping, and they're not driving the symptoms, they're not driving the fatality. So, he put on this graph when you should be using particular therapeutics. And vaccines, monoclonals, antivirals, all early on. And then your anticoagulants, immune modulators, et cetera, later, much later.
That was a few weeks ago. I was talking with David Ho about his work with monoclonals. And they had started with isolating neutralizing antibodies from patients who were sick, and they got a variety of them. And the one that was the star performer, the patient who produced the most robust neutralizing antibodies, and they put this into their monoclonals, was extremely sick. Very, very rough shape and on the ventilator, and clearly, she was able to produce massive amounts of highly neutralizing antibodies, and
too late.
It wasn't fixing anything.
The horses were gone.
So
I'm surprised that she made a good antibody. Brianne, remember that paper where the germinal centers go away in really sick patients?
Yeah.
Yes.
That is so dramatic.
Those antibodies they're made, but they're not very good quality, right?
Right. And, we'll have to see what goes on with the memory cells that are making them. And we'll get more into this today.
It's
going to vary from patient to patient, but that is a really interesting, troubling phenomenon going on.
Well, I mean, it's never 100%, so there are going to be some people. But in that paper, which we did on immune, without germinal centers, you don't have somatic hypermutation and nice maturing of high affinity antibodies. So
Listen to you, a virologist, immunologist. Woo, I love it. Say that again.
I have been teaching that for 10 years, you know. If anything.
Well, I'm slow to get things. You remember that.
I didn't say it. I didn't say it.
If you look at the data in that paper, there are images of the germinal centers, and they are quite dramatic.
Yeah, very nice.
So if you want to know what this looks like, figure 1 of that paper tells you everything you need to know.
Right. The only connection to that statement that you just made, Vincent, that I can top you with is that I once shook the hands of Gus Nossel.
Very good. We had on TWiV, Elio did an AP complexin paper, Dixon.
No kidding. Which one?
Well, it was a structure paper where they're trying to figure out how these things glide, because they used motor protein based gliding. So they're doing structural. Yeah. So they talked about both malaria and tox. I think the paper was malaria proteins.
Okay.
But he said, I shouldn't be doing this, but I really thought it was interesting.
No. The more the merrier, actually.
So was he being a parasitologist without borders?
Ooh.
Yeah, he was, which we encourage, right?
Yes.
Right. Yeah, no one lived there. That's the no borders part.
All right, we have a couple of interesting papers to go through. The first one is published in PLOS Pathogens. It's actually an opinion piece, but it has some interesting tidbits in it. It's called Rapid Deployment of SARS-CoV-2 Testing, the CLIA Hub, with 1 billion authors.
Yeah. This is the first one.
And a lot of people I recognize. The last author is Joe DeRisi, and he is at the University of California, San Francisco. He has been on TWiV, of course, a long time ago. And this is all about a university becoming a diagnostic lab. You know,
and it's marked as an opinion piece, but it's really, it's basically factual reporting. They're telling their story.
Right.
Yeah, it isn't really an opinion, is it, I guess?
No.
There's an
accompanying... we did this. Here's what we set up.
It's their opinion.
There is an accompanying paper that gives you protocols if you would like to try and do this. What's the saying? Do not try this at home. Well, we want you to try this in your lab.
Please try this at home. That's right.
But the point here is, and this we knew from day one, there's a lot of testing capacity in university research labs, right? But they are not clinical diagnostic testing labs. There are big differences, all sorts of regulatory differences and quality control and so forth. So this group of people said, let's see if we can overcome this. And so they partnered with the Chan Zuckerberg BioHub, which is a laboratory, supporting by that foundation in San Francisco. And they completely turned them over to doing diagnostic tests because there was a need for this in the San Francisco Bay Area. And so this was done with BioHub people and also UCSF grad students, postdocs, and faculty. And so it's called CLIA Hub. And the goal is to give free, rapid, high-capacity testing services to local hospitals.
Right.
And CLIA, people have probably already heard many times on the show, is the law that regulates medical labs.
Clinical Laboratory Improvement Amendment approved laboratories, right. And so this BioHub is a nonprofit institute, 110 people, half of them working on infectious disease research. On March 12th, they were organized. This is funny. They say, it allowed frictionless redistribution of personnel into COVID-19 work. I don't believe it. There's someone had to complain.
Yeah, I don't know if it was frictionless.
It takes you back to the gliding thing about epicomplexus.
Yeah, you're going to go to that lab. I'll just touch you and you'll...
Let's drift on over.
So March 12th, they reorganized into this, everybody doing this diagnostic. They developed the assays, and they had to get CLIA compliance, right, which they did. And then they started working, and they have a detailed workflow of what they did.
They returned their first clinical lab results 8 days after they started the project.
That's amazing, actually.
Yeah, but they had been working on similar projects for 4 years before that, right?
Right, but it's it's difficult to overstate the magnitude of the difference
I got
between,
you.
I mean, just the regulatory environment and the procedures, and you have to have everything documented, and you have to follow that documentation, and the amount of training that people are supposed to have to be to the point where you're turning around your first tests a week later. I mean, these folks really buckled down.
So they used nasopharyngeal or oral pharyngeal swabs, and they had a whole procedure worked out, which is, barcoding samples, distributing them into 96 well plates, using automated processes, high-speed liquid handlers extracted, did the RT-PCR with 2 primer pairs, one for the N gene and one for the E gene. The E gene, the other glycoprotein that's completely ignored in the poor virus, but everyone focuses on spike, but it's there. And then they track all this information by computers, of course. And as Alan said, they got their first result 8 days, and now as of October 17th, they've returned 151,795 results.
Exactly.
36 hospitals, jails, nursing facilities, and public clinic have utilized this for testing. And if you look at the map, it's really all over.
Yeah.
Except, I guess, the southern part has been ignored there.
Right, but they're probably they're certainly served by other labs down toward the Los Angeles area.
So they average about 1,300 samples a day, turnaround time 24 hours. But what I find, so that's amazing that they did this and it's ongoing. That's great, and this shows you that if you want to do something, you can do it. But I wanted to explore some of the results which they share. They have a very broad range of CT values that they find in all these samples, ranging from 7, which is a lot of RNA. Right?
Yeah.
The lower the CT, the more RNA in your sample, to 45, which is very, very little.
It's a plasmid.
My CT value was 34.7, and nobody was worried about that. But here, 45 is really nothing to worry about. Now, they say their samples are considered positive if the CT for both primer pairs were less than 40. That's a little high, I think. I mean, we're thinking about 34, 35. If only one primer pair worked, no good. Do it again. And mine was only done with one primer pair, which I think is not good. I think you need to use 2 primer pairs. It really improves.
Absolutely.
Sure.
And then they said 9% of the cases, only one of the 2 probes worked. So they say, suggesting that assays that rely on detection of a single gene alone may have a significant false negative rate.
Right.
And I would say it would also give you a false positive rate, right? Because they have a definition here of positive, meaning both primers work, right? But if you're only using one and it works, but at a high CT, that could be a false positive, as evidenced in my case. So
Vincent, how complicated would it be if you added a 3rd primer?
It's not complicated. It's just a matter of money, how much you want to spend, right?
Yeah.
So, but wouldn't that seal the deal, though? If you were positive for all 3 primers, that's got to be a positive.
2 seems to be enough.
But they were only 80% positive, though, right? That test was only 80% predictive.
No.
Which one?
Is that the wrong paper I'm referring to?
Yes, you're thinking of another paper. There's no.
All right, I'm sorry.
This one is good. They only had 9%. Where only one 91.
Right,
Look, the key here.
Those are in cases where only one was positive. So
exactly, and
their standard was set that 2 had to be positive. So their false positive rate or false negative rate, they don't actually break that down, I guess.
Okay.
Yeah, but
this looks like a very good test.
I think this is the key here: is you need to use 2 primer pairs, and if you get this 9%. One fails, you just do it again. You have to be able to do it again. And that's not hard, right? So the next day you come back and you get a new sample and do it again.
But they didn't keep track of whether the nasopharyngeal or the oropharyngeal route was a better way of obtaining the sample to begin with.
They may very well
I
have.
didn't
They
see
didn't
that
may.
data
here.
separated out.
Yeah, they
What's
didn't
that?
tell us. Yeah.
This is a very short description of the project.
Right. So I would love to know whether one was skewed because they didn't get enough sample.
If you remember the paper we did Wednesday, well, Brianne and
That was
Rich,
so long ago.
That other day.
that was a series of isolations from an immunosuppressed patient who was infected for many days over and over again. And they had both CT values at each time point that they took,
Yes.
which is great.
I was interested that in that. See, because since one of these probes messenger RNA that could be, well, the CT values were the same or similar for both probes both times, indicating to me that at least under those circumstances, the concentrations of the nucleic acids, be they messenger RNA or genome, for both were similar at both times. Mm-hmm. I had been under the impression that they could be quite different, in particular, since one was for a subgenomic RNA, which I was under the impression could be more abundant. But at least in this patient, and for genomic RNA, they would be the same all the time. So I don't know what they're actually measuring, but throughout the course in this patient, both had about the same CT values.
Anyway,
This I'm amused by the March 12th start date for this.
mm-hmm.
It was March 11th that the WHO declared this a pandemic.
Right.
The day prior, and March 12th, the U.S. declared it a national emergency.
And so that's when they had their first sample, you say, March 12th?
That's
Well,
when
when
they
started this project.
That's when they started the
All right,
project.
so as soon as it was a pandemic, they started. Okay.
As soon as it was a pandemic, they said, okay,
All
we're
right.
doing this.
All right. So that's pretty cool. It's open access. You can check it out. And there's a methods article if you want to do this.
Yeah, it's a nice way for those of us who have these types of skills to potentially feel useful during this time.
We just got an email from the dean saying you can't do your own diagnostics in your lab.
Oh, wow.
Really?
Mm-hmm.
And is that for
Probably
legal
liability
reasons?
purposes.
Yes. Liability?
Yeah.
Sure.
I think I mean, if you're going to do yourself and not tell anyone, that's fine. So I could develop a PCR test, and if I'm positive, then I'll go somewhere and have it redone.
Yeah.
Right.
And I wouldn't do Rich and say, you're positive,
I wouldn't believe you anyway.
Right.
You shouldn't. You shouldn't.
Nor should you.
But,
I recall that your old alma mater, Mount Sinai, developed their own test, and it was absolutely better than everybody else's test.
Well, maybe you're thinking of the antibody, the serology
the
test,
antibody test, but that was still another test.
which is very good.
Why can't you develop tests for things if you've got a laboratory?
Well, you can develop them, but you can't
and
use
then
them
get them approved.
to inform people of a result because you're not a CLIA approved
Right.
test.
It's an issue not just with CLIA, but also with a more important law that's abbreviated CYA.
Yeah. I mean, you can think about all of the types of quality control that would need to go into a test if you're going to actually give someone medical advice.
Yeah.
I do
which
remember.
is not what my lab is set up to do.
I do remember a letter from someone who said they set up their own test and they were testing people in their group, whatever that was. And if they got a positive, they would say, you should go see a physician. They didn't tell them what, right? So that is all you can do.
Yeah.
All right. Speaking of Mount Sinai, all right, we have a series of papers, a whole flurry of papers on antibody levels came out. Mm-hmm. And I thought we could kind of go through them quickly. Quickly is in the TWiV sense.
In the TWiV sense, yes. We'll be done before dinner time.
No, we'll be done at 3. These are going to be coming out over the next months because we'd like to know how long antibodies last, right?
Sure.
And if they're protective at all. We see protection in the population and so forth. So a few came out and I thought we could chat about them. The first one is from the Mount Sinai group. Robust neutralizing antibodies to SARS-CoV-2 infection persist for months. This is in Science, and it is the group that includes Viviana Simon, who was on some time ago to talk about this.
Yes, that's
and
right.
Florian Kramer, and who I always confuse with the downhill skier. He was also on TWiV, and he said it would be fine for me to confuse him with, what is his name? Heinz Kramer? No, Franz Klammer, right? That was the skier.
That's right, Franz Klammer. That's right.
Got the same
It
initials.
was not on TWiV.
No,
Shall we get him on?
Sure, why not?
Good.
He's more likely to have a frictionless entry to the show being a skier.
this is all true.
All right, so Mount Sinai, very early on, they developed an ELISA and neutralizing assay to detect antibodies. It's called the Mount Sinai ELISA. High sensitivity, 92.5%,
Right.
and 100% specificity, positive predictive value of 100%, negative predictive value of 99.6%. And Viviana actually talked about all that when she was
did. That's
on the
right.
show. Now, this study actually comes out of a program to recruit donors for convalescent plasma therapy. So they say, okay, you've recovered from SARS-CoV-2. We want to get serum from you and check your titers to see if it's suitable. And so they screened 72,401 people, and 30,000 of them were positive in this ELISA assay. And that's defined by antibodies to spike at a certain titer, or 1 to 80 dilution or higher. So they tell us the range of titers that they find, and it ranges from 1 to 80 all the way up to 1 to 2,880. So that's the dilution at which you lose reactivity. And they basically,
I think an important component of their recruiting in this is that these are people who have been sick and recovered and have antibodies that are included in their test.
Mm-hmm.
They do not include people who were just PCR positive, but not necessarily really sick. The population's a little different, okay? And I think is less likely to, relative to other studies that we're going to come up with, and they make this point themselves, less likely to include people who had extremely mild cases.
that's right.
Right.
Yeah.
But at the same time, they also don't have a ton of people who had extremely severe cases either.
Right.
This is a mild to moderate cohort.
Yeah,
Yeah.
that's right. And so again, these are ELISAs just for antibodies that bind the spike, right? They're not yet neutralizing. We'll get into that. So they conclude that they say the vast majority of positive individuals have moderate to high titers of anti-spike antibodies. Okay. So they actually went through all these 72,000 people and measured each one in this assay. All right. So then they say, what about neutralizing antibodies? And they use infectious virus for this. So, you make dilutions of serum, you mix them with a fixed amount of virus, and then you do a you infect cells. You could, in my lab, you would do a plaque assay, but you don't have to do a plaque assay. You could just look for virus killing of cells and so forth.
CP, sure.
And this is the genuine article. These are not pseudotyped.
That's right. The genuine.
Virions, right?
This is the real deal.
Real SARS-CoV-2.
Yeah.
Authentic, as they would say.
Authentic.
I heard someone calling a pseudotyped virus a SARS-CoV-2 entry virus online, and that made absolutely no sense. And I was sort of like, what are we doing?
Yeah.
I would call it a sheep in wolf's clothing.
Yes.
Sheep and wolf. Yeah, that's correct. You
Yeah.
got it. Anyway, so then they took 120 samples for their newts. And therein is my issue with this.
No, newts are not their experimental model. He means neutralizations.
Yeah, the neutralization. Sorry, I like to abbreviate. It's a cool
Turn me
word.
into a newt.
Newt is a cool word, right?
Yes, it is.
He's turned me into a newt.
And I'm sure there are viruses of newts
better.
as well.
Yes, I'm sure there are viruses of newt, but this is not one of them. So they
So
looked
should
at
we go
neutralization.
down that rabbit hole for a little while?
Well, it's,
Yes. No.
They looked at 120 of the 30,000 that were positive. They took ones that ranged from no antibodies all the way up to 2880. Okay. And they find neutralization at different levels. They correlate more or less with the rough antibody titers determined in the ELISA. Okay. But it's only 120, and I would argue that this may not be representative of the 30,000. And the reason I say that, I'm trying to understand why this paper is really different from the other two that we're going to talk about.
Yeah.
You know,
I have a lot of thoughts about that.
we'd love to hear them.
Yeah.
All right,
In fact, reading this paper and the Nature paper, I have been formulating a class activity and assignment immunology class.
Yeah, that's good. Explain why these are different. Yeah. And then when you get the best answer, you could share it with us.
Perfect.
Just write a letter to the editor of Nature. Hell with that. Sign the class of.
So, all right. So these 120 have a range of neutralizing activity. You know, they have low to medium to high neutralizing activity. Then they say, how long does this last? Can we bring some of these patients back at different times and measure it again? So they recalled 121 of these people. You know, so we have that first titer, then we have 52 days later, which is, there's a range, obviously. You can't get everybody in at 52 days. And then, so the second time point is at a mean of 82 days post-symptom onset. And then the third point, 148 days post-symptom onset. All right, and they see drops in antibody titers, which you expect. And they find that if you started out with a low titer, then you are more likely to go to undetectable levels.
So for the listeners, let's qualify that drops in titers, which you would expect.
Yes.
Okay. Because what happens is you mount an immune response, you get a nice boost of antibody, and then that naturally, under virtually all circumstances, correct me if I'm wrong, Brienne, that wanes down to some steady state level that's above background, okay, but not as high as it was. Exactly. Because you don't want to be, you don't want or need to be totally blown out with antibody the whole time. So that initial decline is expected.
Right.
Yep, exactly. So when I look at these declines, I see that there is a decrease in antibody titer, but that the antibody titer remains measurable at all time points. It never goes all the way to undetectable or below the background. And they do see, statistically significant differences between the time points, which is not what the title says.
Yes. It's exactly
picky.
No, I agree completely. The title is not really appropriate.
The title giveth and the paper taketh away.
Exactly.
Exactly.
Yeah, no, the title I think is very hopeful.
Yes.
Yeah.
But we don't need hope. We need truth.
Yeah.
Right. And so the title says robust neutralizing antibodies to SARS-CoV-2 infection persist for months. Mm-hmm. And if we take that as meaning persist as being measurable or persist over background, then that is true. The level, however, does not stay the same. The level decreases over time.
Sure.
Right. Now,
And if you interpret robust as modifying neutralizing, then it's okay. But if you interpret robust as modifying the level, then that's not what's happening. The level comes down.
I'm in favor of not having words like robust in titles of papers.
Adjectives. Yeah.
I think
agree.
those are, your interpretation of the data. They're not factual representation. So I don't think that's
and by the way, these are not like as in journalism where somebody can write an article and then that's a perfectly reasonable article and then some editor writes the headlines. Okay, that's not what's happening here. These are the scientists who are hyping their work with a title.
Yeah, I agree. Now, the key here is that they see up to day 148 antibody levels. At some point, they're not high, but they've gone down, but they're still maintained at 148. And they're claiming that this is the product of long-lived plasma cells in the bone marrow, which memory cells, right? And I don't think they can make that claim at all. What do you think, Brian?
They can make that claim, but they don't have any data to support it. They didn't. Sure. There are no bone marrow biopsies. There's no isolation of that.
So, Vincent, is it possible that they're reacting to remnants of viral replication? little bits and pieces that are thrown out into the bloodstream as the cells disintegrate that fail to produce a whole infection. And the spike is over here and the capsid, other proteins are over there. And it's just percolating along, sort of keeping the immune system alert to the fact that you've been exposed to this.
It's not how we think it works,
you often said, sorry?
It's not how we think these infections work, right? If you look at Daniel's...
I thought you said when they do a test in the blood for the organism and they only use a single probe, it could be just remnants of RNA that are floating around, just bits and pieces rather than the whole virus.
Yeah, but... So yes. So here's the thing.
It's going gangbusters outside my window, sorry.
So nobody has found... In all the reports, there's no infectious virus after 20 days, okay? But there's PCR positivity for sure. But if you have little bits of virus, I don't know how they would keep stimulating the immune response, right?
Brianne?
I mean, I could imagine that there could be some bits of virus still sitting in a follicular dendritic cell in a germinal center, if there were, well, put together germinal centers. But I'm not sure that going out today, 148 is long enough to tell me whether there are particularly long-lived memory cells, or whether these are just cells that have not yet declined all the way.
They haven't gotten around to dying.
Yeah, that's what I wanted to hear. And I also am interested, Brianne, in what sort of levels above background of antibody one might expect for an acute infection at this time, or even, another year out. Okay, you can detect whether somebody's seropositive for an infection a long time ago, many times, but how much antibody are we talking about?
Right. I don't have a number for you in terms of a specific titer, and I think that varies quite a bit from virus to virus. But I think that you would probably be getting closer to that 160 to 80 range, above measurable, but not this high above background at a year out. I think here we're on the way down to that.
Yeah, so something detectable as opposed to zero, like even 1 to 4. So for polio, 1 to 4, 1 to 8, 1 to 16, you can find serum antibody levels years after infection. So it's not zero. So I agree. It depends on the virus as well.
And, years after the infection, even a low level positive, I would interpret as evidence that there's some memory percolating there in the background, and that would make me feel safe.
Right, exactly. Because then you're getting out past the half-life of any individual antibody, and that antibody must have been newly produced by a cell.
That's right. Now, Stanley Perlman looked in SARS patients 15 years after their infection, no B cells, no memory B cells in the bone marrow. They ain't there. They can find T cells, virus-specific T cells, but not B cells. So for some viruses, they don't endure. And
for the seasonal coronaviruses, people seem to be able to get those again, right? And do we have antibody data from those?
Yes, remember we did a paper where in the Netherlands they looked at an AIDS cohort, right, where they had banked sera for the last 40 years, and the serum levels go down, but they don't go to zero; they go down, very low, and then people get reinfected and they go back up, and then they go down. So there is always some level of antibody present.
Yeah, and so the level that is detectable and the level that is protective are not necessarily equal.
Right. And the level, that oscillation that you see in the immunosuppressed patients implies that they don't have sterilizing immunity. They are getting infected and they're mounting an immune response, but it's probably protective of getting symptoms.
So I'm glad you brought up that word sterilizing protection, because in contrast to what we're hearing right now in the parasite world in eukaryotic cells, particularly for an organism called Toxoplasma gondii, you can remain positive for most of your life because the organism survives in a small pseudocyst in your body for years. And every now and then one of them breaks down and releases some antigen, and that sort of keeps the immune system alert to the fact that you've still got a pathogen lurking in your tissues somewhere. So are we sure that when someone recovers from SARS-CoV-2 that there's no persistent low-grade infection going on that you
Sure
can't
is a very
demonstrate by
strong word.
ordinary technologies, but could that account for this result?
This paper we did Wednesday is the first case where we see an immunosuppressed person where he's constantly reinfected with his own virus. There's a little bit in him that every time they stop the therapy, the antiviral therapy,
exactly.
he gets another infection. So that's the case, but there's no documented in healthy patients of that kind of thing happening.
No, but what about people that are subclinical for immunosuppressive events? You wouldn't notice them because they don't get sick from them.
It's possible. But
we're not a single clone of people.
Look, with Ebola, remember, we thought it was an acute infection, came and went. Then all of a sudden we found people who were infected for months and had a recrudescence of their original virus. So it can happen. We haven't seen it yet for SARS-CoV-2, but it certainly is possible.
Well, we thought maybe some of these reinfected people might have been infected with their own virus, but the virus had mutated a little bit since the first infection, so we could prove that they didn't get infected with their own virus.
That's right.
Right.
That's right.
And that patient we talked about on Wednesday was pretty severely immunosuppressed.
Yeah, that was a real special case.
That's a very unique case.
Yep.
Right.
So the last thing I wanted to mention here, they have a nice discussion of correlates of protection. And Matt, we've talked about this, but the correlate of protection is something you can measure, like antibody or T cells, that gives protection against infection. And they say these are usually based on a specific level of antibody made by vaccination or natural infection. They give some examples. All right, numbers. We love numbers here on TWiV, especially with dollar signs in front of them. So, for example, a hemagglutination inhibition titer of 1 to 40 for influenza virus reduces the risk of getting infection by 50%. So hemagglutination is a way of measuring the number of particles, and you can block it with antibodies. And they say similar titers have been found for measles virus, 1 to 120, hepatitis A, hepatitis B, and many others. So that's good because then you have a goal to reach with your vaccine. You say, if we get to this titer with our vaccine, we suspect you'll be protected, right? And that's the goal for SARS-CoV-2. We don't have those correlates of protection in humans. We have them actually in non-human primates, as they point out, but not yet in people.
Now, you don't actually need those numbers to develop a vaccine.
That's right.
But longer term, after, in this case, hopefully we get a vaccine sooner. Longer term, I hope that we're going to be doing these studies and tracking immunity levels and you know, whether people get infected and what's their antibody titer. What is it? Is it 1 to 120? At what point do you lose protection? And then, if necessary, you can use that in refining your vaccine or in choosing a vaccine design, since we're probably going to have multiple choices in that regard.
Yeah, no. Right now we do preclinical studies in animals. We get something that protects animals, and then we do clinical trials in people, and hopefully they work. And then, as you say, we can study them.
And work in this case is people are not getting hospitalized, basically. It's we're setting the bar pretty low.
All right. Let's go through the others a little more quickly.
Yeah.
Yeah.
Okay.
Well, I think we've done a lot of the background discussion here, so we can.
The Nature Microbiology paper, longitudinal observation and decline of neutralizing antibody responses in the 3 months following SARS-CoV-2 infection in humans. Now, that's a neutral title.
Yes, that's a neutral title.
And in some ways, that title could describe the data in the other paper.
Yes.
This is from a variety of institutions in the United Kingdom. And again, there are many authors on this paper. But again, it's looking at antibody responses in patients. In this case, the cohort is 65 individuals with PCR-confirmed infection. And they also did a separate one in 31 seropositive healthcare workers. And they say the full breadth of COVID-19 severity from asymptomatic to severe respiratory failure. And that's interesting because it turns out the people with the more severe disease actually make higher titers of antibodies, although I would guess they're not very good, right? They're probably not going to last a long time.
But as we were discussing earlier, this is I would say a significantly different population.
It is.
Of individuals.
Yes.
So they again they do first ELISA, just antibody binding. They look for antibodies that bind to spike, the whole spike, the receptor binding domain, and the N protein separately by ELISA. Basically, all but two people make antibodies against these antigens. They also looked at over time, but I'd rather do the neutralization. Because next they use the neutralization assay. They did both the serotype and some with infectious virus to confirm it. And again, they see a range of neutralization potencies over.
And they're doing this neutralization assay is with pseudotyped virus.
Yeah, and they did confirm some of them with infectious virus, yes. I believe, right? Yeah.
I think so.
So the real key here, so again, many patients make neutralizing antibodies of different potencies, just like the other paper. The real key here is the longevity. And here, they say in all cases, the neutralizing titers decrease. And for some, they go to undetectable, and those are the people with the less severe disease. And people with more severe disease, they maintain titers longer. And this is the case where they measured them with infectious virus just to confirm their finding.
And they aren't going out as far. They're only going out about 70 days. Is that right?
Yeah, that's 70 days.
That's
right?
correct.
Yeah.
That's right. But they're not.
And that's days post onset of symptoms, not necessarily days post. Well, it wouldn't be post infection. So this is a little bit of a lag.
So if depends on the antigen. The graphs are informative. You can see the clear decline with time up to 80 days after infection. And depending on what you're looking at, you can see different levels. In no cases is it zero yet. I guess there's just, there was one there.
Some of them get pretty low.
They get low, but not zero.
So for me, looking at this, it didn't look a whole hell of a lot different than the paper we just did.
No.
People get an immune response. It wanes as it should. Most of them maintain it for a period of time. If there's any difference, it's that people who basically had asymptomatic to very mild disease, who were a minority, if included at all in a previous study, may wane to something closer to background, but they weren't much over background to start with.
Right.
Okay.
Yeah.
So
now they say, and this is important, the kinetics of the antibody response in SARS-CoV-2 is typical of an acute viral infection.
Bingo.
At so many days after infection, you get a rise, and then it slowly goes down. And they say here, further follow-up is needed to determine whether the antibody decline will continue on a downward trajectory or whether they will plateau facilitated by production by memory B cells. So they haven't gone out long enough to do that. So Brianne, what would you say is the length of time you need to follow to really know, without doing bone marrow biopsies, whether you've got that low titer? How many months, you said?
I mean, I'd probably want to go, greater than 6 months. I might go for a year. But certainly a bit longer than this. And Rich, I agree completely. When I was looking at these papers, in a lot of ways, I saw that the very big picture was that they were seeing kind of the same thing.
Yeah.
Right. And something that's pretty standard for an acute infection.
Yeah. Exactly.
Yeah. And they've just not gone out.
To me, this was all good news.
Well,
essentially.
But it also, to me, one of the things I take away from this is that there's kind of a Goldilocks situation here where if you are asymptomatic or really minimally symptomatic, that's great. You didn't develop bad disease, but you also didn't really develop much in the way of antibodies. So you're probably not, if that's the correlative protection, then you probably didn't get anything out of it.
Yeah. Right.
If you developed some disease and got over it, then you get this spike and it lasts for a few months and it's declining and we don't know exactly what that means. If you got severe disease, of course, then that sucks because you got severe disease. So
The last thing in this paper, they have a discussion which is useful. They say, well, what are we going to need for protection of people after a vaccine? We don't know because we don't know what titer. I mean, so these low titers we're seeing, is that enough to protect people? We have no idea.
Are the antibodies even what's really going to be protective, or is there a T cell story that we're not looking at?
Well, yeah, that's the other, what do you say? The elephant in the room, right?
Yes.
Yes.
But it's not actually the low titer that's protecting you. It's going to be the memory B cell response that's going to protect you, that's going to kick in,
Yes.
right? The low titer antibody is just a sign that those memory B cells are present, right?
Right.
Is there a way of conducting a study of people who have already caught it, who are obviously thinking it's now free to move about the cabin, so to speak, to see whether or not in 6 months' time, any of those people get reinfected?
I'm
Well,
sure
we've
it's
already
being done.
got several reports, reliable reports
Yeah.
of people getting reinfected.
We'll
No,
see.
but
We'll see
not at the
more.
level that we're looking for, I think.
No.
Right. Well, the thing is, I mean, I look at those and I say, okay, so there are a few reports in the literature of people getting infected. And on the one hand, people could say, yeah, but it's only been seen a few times. But it's only really been looked at a few times, which to me strongly suggests that that's going to be maybe the rule rather than the exception. Because what are the odds that the 3 people in the world who got that rare event happened to be the ones who were thoroughly analyzed in scientific papers in a pandemic that's been running less than a year?
I think we need to move 6 to 12 months out, and then we'll see, because that's
Yes.
with the
Yeah,
common
yeah,
cold
exactly.
coronas. 6 to 12 months is when you see the reinfection. So we just haven't been long enough. But I think, Dixon, people will be looking at that very carefully.
Sure.
Anyway, the point
And
that
especially post-vaccine,
exactly.
that's going to be the real test
That's right,
when
that's right.
because at that point, people will be, and I will be presumably saying, okay, now I can go out and play cards with my friends. And we're going to hopefully keep track of how well that works out.
You still have to wear your mask.
One last point. So they say in the discussion, in rhesus macaques, where they've immunized, they develop neutralizing titers of 100. They are protected against challenge, 35 days after the immunization, but they still have virus in the nasal swabs, much less. And they say this suggests not sterilizing immunity, but immunological control. And then they cite a second study where the non-human primates were protected and with neutralizing titers in the 8 to 20 range, which is quite low, actually.
Yeah.
And so they're saying, what are we going to expect from a vaccine? If the vaccine titers are low or they decline, we may need to boost periodically with some of these vaccines to keep the titers high enough.
It may be get your flu shot, get your COVID shot.
Every year, maybe. Yeah,
that's
it could
right.
be.
That's right. That's right.
Or it could be that you'll get your immunization and get boosted by the virus.
By the virus that's
Also
spreading
very
because
possible.
people don't have sterilizing immunity, so they're spreading the virus around and it just becomes endemic. Yeah.
So what you need, it's like what you need is some immunological capital. Okay.
Yes.
And once you have that, you're okay.
Yeah.
And that's true of a lot of infections and a lot of vaccines. Today.
Absolutely.
Yeah.
Absolutely.
All right, the last paper is a medRxiv preprint, but I've been told by one of the authors that it's been accepted. I think JID, I don't remember what she told me. So Theodora Hatzianu and I were having a conversation about these other papers, and she said, well, we have a paper that's coming out. So she sent me this, and this is from the group at Rockefeller, and I believe we did a paper of theirs earlier on similar studies of antibodies in the blood of patients. And so Theodora and Paul B. Nash and others from Rockefeller, and they have collaborators as well.
And Theodora and Paul have both been on TWiV.
They have both been on TWiV. I invited Theodora today, and she said it wouldn't be fair unless you have Franz Klammer on. No, Florian Krammer.
But it's all downhill from there.
Okay, she said you should get us both on sometime. We can debate this. Okay, sure. And so here they have a cohort of patients as well. The main point of this paper is actually to compare different point-of-care diagnostic, serological assays from 3 different companies. I don't really want to talk about that, but they look at longevity of these antibodies. So they have this cohort is how many people? 97 people, none of them hospitalized. So this is mild to moderate illness. And they again look for antibodies to different proteins, and they do some neutralization assays. So let's look at that. Go right to the videotape. Right, Dixon?
Let's go to the videotape.
Got it.
And they have time points. What are these time? Oh, this is the paper with the wonderful graph. Figure 2. For those of you listening, go download this paper when you get home and you're not driving. This is
no, wait a minute. Are you talking about part A?
Yeah. That's a stratification. That's actually.
Oh, that's the stratification.
That's stratification. Yeah. But I still like it.
The x-axis is just individual participants.
Those are participants. Yes. Yeah. So on the left, we have people with high titers, and on the right, low titers. But you can see with each subsequent visit, it's going down because the visit 1 is red, visit 2 is green,
okay. Yeah.
visit 3 is blue. And then when you go all the way to the right.
D,
sorry?
Part D is where you see that everybody's got a declining curve.
Yeah. That's, and some who start high, they don't go to baseline, and the ones who started lower, they go to baseline. So basically similar results as the second paper, right? But on the stratification, it's interesting because there you can see the ones with the low titers to begin with all the way on the right, they don't have far to go to be nothing, right? And so again, this consistent with it's a response to an acute infection, you have antibodies produced and then they decline. And they're not claiming that...
Underscoring the point Brianne was making about everybody, mounting a unique response. You've got a couple of little blips where visit 2 or visit 3 is way up above the line.
Yeah.
So there's one in every crowd.
And they say we don't know if these are going to keep going down. We don't know if there's memory. We have to go out further to do this. But in all 3 papers, there's this typical acute infection, a rise and then a decline. And how far the decline is, we need to see.
We don't know yet.
I'm hoping that we're swamped with vaccines that are so successful that we'll never know the answer to this.
I use this kind of data to suggest that nothing in this world will kill everybody because mostly we'll survive because we've got a robust immune system.
Dixon, that's why I emailed you the other day to ask about the crows. In West Nile.
That's right.
Okay. Because
they're doing great now.
Nothing is going to kill everything.
They weren't to start with.
No, of course not.
90% of them, right?
Listen, that happened when the Spanish came over to the New World back in the 1400s. Can you
imagine a pandemic like that?
No, this is nothing.
But we don't have to. Yeah. We're living in one.
Well, no, this is not 90%.
No. That's I take that back. But if you had lived in the New World during the 1400s and 1500s, and you had never seen any of those infections before, then that would be the equivalent.
There's some things that can kill everything. You said there's nothing that can kill it, but there are some things like nuclear holocausts, right?
Yeah, and the conquistadors also showed up with bullets, which were very lethal.
Yes. Any place where we're thinking about the diversity in genes of your immune system, then yes, in general, things are not all going to kill you. However, nuclear holocaust is not really.
No, I draw the line.
Once again, physics is non-negotiable.
Your immune system is not going to.
No, it's not going to do it.
So one of the things about this paper that I found interesting and I wasn't aware of is that some of these tests rely on looking just for antibody to nucleocapsid protein.
Yeah, that's right.
And some rely,
it's an indication of infection.
Thought, Spike is the obvious guy that I would, I don't understand really.
Well, you're making antibodies to many viral proteins. So if you just want to know if there's an antibody response, not a neutralizing response, right? Just bulk antibodies, you could do nucleocapsid is extremely abundant. That's why it's a good one.
Right. Yeah, I guess so. Yeah. Okay.
Yeah, I thought the part at the beginning about the variation between tests was quite interesting.
Yeah, there is a variation. It actually changes with time and infection as well, right? Just curious.
And so can you summarize that, Brianne? How does that go? I forget.
So basically they use different PCR tests at different times and actually check to see whether or not patients are testing positive and look at the sensitivity of those tests. And so this is figure 1. where they're looking at tests from 4 different companies. And they can actually see that the tests have very different sensitivities at different times.
Are you talking about antibody, not PCR?
Yeah, you said PCR. You mean antibody?
I do. Yes, sorry.
Okay.
So they use the antibody tests, and they see very different sensitivities over time.
Yeah.
And so, for example, 3 of the tests have an over 90% sensitivity at the early time points, day 21 to 40, and 2 of those stay above 90% at late time points, and 1 of them is down to about 70% sensitivity.
Yeah, that's weird.
And so I think that's really interesting, and something that we should all be taking into account as we read the other papers is what technique are they using.
So they say
there's one that it's not as dramatic, but it starts out at like 85% sensitivity, and then over time it goes up to 90+% sensitivity.
Exactly.
So they actually say these different tests are more or less well suited for surveillance of populations.
Yeah,
these are not meant for diagnosis.
Yeah.
Okay. Because of these issues, they're not. But you could do them for population surveillance. You would say, all right, how many people in New York City have been infected? Right. You could do that. Okay, let's do some email. I was only 15 minutes over. Not bad. 14. Rich, can you take the first one?
Sure. Li Ping writes, Dear TWiV, the Chinese vaccines will soon be available to us at a very reasonable price. So I assume she's writing from China.
Yes.
Compared to that of the flu vaccine. As we make the decision on whether or not to inject this thing, I'd like to seek your input.
This thing.
My understanding is that this vaccine is live attenuated vaccine. I'll come back to that because that's not the case. Made with 1930s technology. Given the fact that coronavirus has multiple strategies to evade the immune system, I doubt the efficacy. What are your thoughts on that? Do you think the U.S. would make the vaccines available to the world once it is cleared by the FDA? Also, regarding the current circumstances on COVID in China, seems that they really have this thing under control. But as the winter approaches, we will see how things change. My hunch is that there's going to be a second wave of this thing here in China. Okay, so the vaccine, she actually sent a picture of this from Sinovac called CoronaVac, and it is a SARS-CoV-2 vaccine produced in virocells and inactivated. Yeah. So it is an inactivated vaccine, not a live vaccine.
It's the real SARS-CoV-2 virus that's been inactivated.
Correct. And I would say, with respect to my opinion on this thing, it has been approved. It has been given emergency approval in China. It is undergoing phase 3 trials. There are no published efficacy data in terms of preventing disease at all from any trial. There are, I believe, published data from earlier trials saying that it will induce an immune response, but that's not the same as preventing disease. And I would say that it is the United States regulatory agencies, the FDA and et cetera, are not going to even give emergency approval to anything that doesn't show any kind of efficacy data in terms of preventing disease. So also, I don't know what the safety profile of this is, okay? There are certainly safety data from earlier trials. They're going to be in a small number of individuals. As far as I know, there's been no data published from the larger phase 3 studies. So we don't know in a fairly large population what the safety profile is. So from my personal point of view, this is not ready for prime time. Okay, but
no pun intended.
It's been no pun intended. It has been given emergency use approval in China. So personally, I wouldn't take it.
Okay, so does anybody know if this is the one they gave to their army?
No, that was an adenovirus. That was an adenovirus.
Oh, that's right.
Different.
That's right. That's an adenovirus one. Okay. So I followed this one toward the beginning of the pandemic. This was one of the first vaccines because it uses the most obvious and one of the oldest methods of making a vaccine. You take the virus, you inactivate it, there's your vaccine. Just dirt simple. We have a lot of vaccines that are made this way that work very well. It's an approach that can be extremely effective. It's a vaccine that would actually have made a lot of sense to include in the warp speed trials to do in the US, but there is a deliberately racist stipulation in warp speed that excludes any product made in China. So we won't get this. It is probably going to be the first vaccine put into wide-scale use. I don't know what the status of the phase 3 trial is. Right. The updates that I've gotten have only been on stuff that's in the US and that's in warp speed. I do know that they are doing a phase 3. I believe that started earlier than any of the warp speed phase 3 trials because this was so fast out of the gate. And I would say, advice to Li Ping, if the phase 3 data are published by the time you're going for your shot and they look good, then yeah, I'd get this. It's Sinovac seems like a reasonable company. And they've taken a reasonable approach, and there's no reason to believe that this wouldn't work. But absent the efficacy data, yeah.
If you want to look at the status of things, you can look. There are a variety of different resources. One of which is the WHO landscape draft landscape document that provides links to the clinical trials, and this thing is listed there. The other is RAPS. I forget. We talked about that a while ago. Let me quickly look this up.
While you're doing that, I just want to point out two things. First of all, both inactivated and attenuated vaccines are 1930s technology.
Yes.
The first inactivated flu vaccine made in the 30s, first attenuated yellow fever made in 1937. Looking at this box is very interesting. First, it says on the box, emergency use authorization. Secondly, it's adjuvanted. It has aluminum hydroxide in it. Mm-hmm. And third, description, the vaccine is the milky suspension.
Yes.
Has Virocells in it too, yes?
Well, it's made in Virocells.
It was made in Virocells.
Well, how much of the Virocell got transferred onto the vaccine?
So there must be some DNA there, I'd bet. Yeah.
So RAPS is the, let me get this for you. The Regulatory Affairs Professional Society. They have a website that I think we mentioned in an earlier TWiV. I think that's where I got it. Or maybe I got it from my vaccine buddies. I'm not sure. At any rate, they have a website that tracks all these, and they give some details. There was a phase 1/2 trial with 743 volunteers. There was and there's a phase 3 trial underway. They say the results from phase 1/2 were posted in a preprint server. So those are available, indicate good safety and immunogenicity. And we don't know anything about the phase 3 trial, but I would say that from the landscape document, you can look up that trial if you want. So that's what we know. Yeah. And it was given emergency use approval in China. So the other part of her question is, will the U.S. make its vaccines available globally when they are approved? And my comment on that is I doubt it until the U.S. population is vaccinated. And I would point out in this regard that there is an effort that I think we've talked about before called COVAX under the World Health Organization, which is an effort a multinational effort to make sure that effective vaccines are distributed where they are needed globally and to the neediest individuals, and that the U.S. is not quite deliberately not part of that effort, given that the current administration is not a fan of the World Health Organization, among other things. Okay, I would hope that if the political climate changes here in the U.S., that whole thing would be rethought and the U.S. would be more willing to participate in a global effort.
With that all said, the U.S. manufactured vaccines are all being made by corporations, private corporations that while, yes, they are U.S. based and are bound by U.S. regulations and will prioritize vaccinating Americans, in their press releases and presentations so far, they have made it abundantly clear that they are all aiming for global markets. And most of them are seeking approval simultaneously in the U.S., the EU, Japan, the developed world, Australia, presumably as well. So there's that. But in addition, I know at least one of them, Sanofi, has the capacity and is capable. This is the company that makes most of the world's flu vaccine every year. So for them, when other companies are talking about, we're going to scale up to 50 million doses a year, they're going to say, that's just, that's November for us. And they are very deliberately scaling up their vaccine, their SARS-CoV-2 vaccine, to produce something like 1 billion doses a year. Now that's currently in phase 3 trials. We don't know. Is that one in phase 3? It's either in phase 3 or about to start phase 3 yet. So that's the kind of scale up they're looking at. The other companies, Moderna, et cetera, which are earlier on, Pfizer, they are also scaling up, not quite to that magnitude, but they're very interested in getting this out to poor countries as well as rich countries.
And just one final note, there are actually 4 attenuated vaccines under development in different countries, according to the Milken report, and they're all being made by codon deoptimization, and they're all in preclinical development. So it'll be some time before we hear about them.
Talking about global reach, actually, I was looking over this stuff the other day and looking over the phase 3 trials for the recombinant vaccines and how they're being done and where they're being done. And I was impressed, shall we say, with Johnson & Johnson's reach, okay? Because their phase 3 clinical trial is happening all over the place. And I assume that's because they are all over the place. Okay?
It's funny because Moderna and Pfizer get all the press because they're first to go into phase 3. J&J is bang alongside that, and they're just quietly scaling up this massive effort.
Yeah, and the groups that they are working with in academia are groups that have done HIV vaccines around the world.
Yeah.
And so it's easy for me to imagine them already having an infrastructure for doing all sorts of vaccine trials in lots of places. The
other company worth mentioning in this, they're actually the furthest behind, but I really want to see what they come out with, is Merck. Mm-hmm. Because in terms of developing a wide variety of vaccines, they're unequaled.
Did you hear, Alan, that they have exhumed Maurice Hilleman for this?
If they could reanimate him, they would have. So it's
Sans Tourette syndrome
that is well established in doing this, and their approach is a very cool attenuated vaccine that I hope works out.
Isn't it a shame that Coca-Cola isn't in the vaccine business?
Why is that?
It would be because they're all over the place.
I was absolutely years ago. I was at a public health conference, and somebody used that in their introduction. He picked up, he brought a can of Coke to the podium.
That's right.
And he picked it up, and he said, if the cure to malaria, HIV, and tuberculosis was to give everyone in the world a can of this,
yes,
we could do that.
That's correct. That is correct.
We could do that today.
Yeah,
that's right. So
now,
I imagine
he was putting that in the context of distribution of vaccines.
Right.
You could
sure. As soon as one of the U.S. vaccines is efficaciously proven, or proven efficacious, I should say, it seems to me that it's an easy license deal to get other companies and other countries on board very rapidly after that, so that you could have, within a year, you could have the whole world making the same vaccine, basically.
If you wanted to outsource that, yes.
Right. With a licensing agreement, that's what that's all about. Right?
I don't know if you should ever put easy in front of licensing deal, Dixon.
No.
Depends on the acuteness of the situation.
Now, I have been accused on the website of skipping over Brianne in the email, which I didn't think I did. So, Brianne, can you take the next one, please?
Sure. Leah writes, "Greetings, TWiVers. I was so excited when you read my last limerick on your podcast that I've been looking for just the right inspiration to send in another one. It also gave me a great excuse to share your podcast with people I know by joking that I'm now a celebrated poet. Ha! Listening to TWiV 674 this morning, I was intrigued by the article you discussed about the effect of SARS-CoV-2 spike on neurons. So without further ado, here is my question in the form of a limerick. Since in vitro spike affects neurons and made rats not feel pain quite so well, might reducing pain in rat toes be what's going on in your nose? Is this why folks with COVID can't smell?" That's very good. I appreciate you all so very much. I love your thoughtfulness and commitment to cutting through all the nonsense bombarding our nation. All the best, Leah, and Leah's from Oak Ridge, Tennessee. And whoever's commenting, thank you for wanting me to read things.
It's interesting. She uses the word bombard, and she's from Oak Ridge, because there is a connection there, by the way.
Yes. Yes.
Unfortunately.
So the question here is basically, is the potential influence on pain mechanistically or related to the influence on smell? Can anybody address that?
So what's happening in the
Richard
smell,
Axel could.
What's happening, he's not here, unfortunately.
No.
What's happening is that the virus is reproducing in the support cells in the olfactory epithelium, not the actual neuron processes that are sensing smell, but the support cells, and destroying them. And that's why you have a loss of smell. Now, whether neuropilin, which is the protein involved in this pain issue, is involved or not, I don't know. But it's an interesting possibility. But they're different pathways. So olfaction is different from pain transmission, right? So it's a great limerick.
Yes.
Yes.
Very good. Very good. High on our list.
Alan, can you take the next one?
Sure. Barry writes, results of Melbourne mask trial. It seems masks have worked incredibly well at reducing the spread of SARS-CoV-2 here in Melbourne. Sure, there were a few small confounding factors that muddied the results a bit. You may remember we were having 300 to 400 cases a day when masks were made mandatory in late July here in Melbourne and Victoria, Australia. To further set the scene, all other states were seemingly virus-free or having single-digit cases, and we Victorians were a little upset at this state of affairs. A border sign leading into Victoria was graffitied so it read Covic instead of Victoria. Very amusing. After masks became a thing, and thankfully adherence was very high, case numbers stubbornly and disappointingly continued to rise. For the next 2 weeks, they rose into the 400s and 500s. I heard epidemiologists explain that the masks were working. It was just that there was a built-in latency in the case numbers. Well, okay, I thought, let's wait and see. We did wait and see, and they rose into the high 500s and up to 600. So we added a few more small additional measures on top of the mask wearing. All retail shops closed, 5-kilometer radius travel restrictions from home, curfew from 8 p.m. to 5 a.m., no gatherings in the home, no gatherings out of the home, limit of 1 hour for exercise outside, limit of 2 people from the same household outside at once, and then only for exercise, limit of 1 person per household per day for grocery shopping. Other than work, only 4 other reasons to leave home: getting tested, getting medical attention, caregiving to others, and groceries. Workers traveling outside the 5-kilometer limit were required to have an employer's signed certificate that they're a permitted worker. This was called stage 4. And then, as you can probably already guess, cases continued to rise for a short while anyway, up to a peak of 700 in early August. And then they finally fell. In fact, they plummeted from 700 to the low teens in less than 2 months. We're now down to a 14-day average of 6, and last week it has only been 2. It's been a hard and grueling lockdown, but we had the other states of Australia to look to and help us. They sent us healthcare workers and contact tracers and test kits. We used the lockdown to dramatically improve our own Tetris, but we are cautious in relying on it, and we are done taking any chances. Although Premier Dan Andrews has stated we're not aiming for elimination of the virus in Victoria, it does suspiciously look like we're aiming for such low numbers that a toddler with a toy phone could Tetris this thing. So whatever happened, however it happened, it worked for now. The masks worked, and so did all those other things. No idea by how much, but the culture here in Melbourne feels different to the rest of Australia, like it's permanently changed. At least we're having some fun with it, too. Can you guess who this is with their new mask on? And that looks like Audrey Hepburn in a billboard. And I would actually say that this is evidence that the masks didn't do a whole lot, but the very aggressive lockdown certainly got the job done. Which would kind of be consistent with what we've seen in other places. I mean, you can't just mask your way out of this. They appear to be helpful, but just saying everybody wear masks in and of itself, if everybody's going out still to restaurants and bars and all the other things, that's not going to be enough by itself. That's kind of asking a lot of them.
And Melbourne is a very bar-conscious city. I lived there for 6 months, and they love to socialize. They love to sing. They love to drink. They love to eat. And it's a remarkable thing that they actually did it.
And remember, when you go into a bar and a restaurant, you have to take your mask off.
You have to take your mask off,
So I think saying there's masking is a little misleading, because in a lot of cases there's not, especially in those cases where we know most of the transmission occurs in these social settings.
Yes.
So, by the way,
this is a serious lockdown.
It is
for a long period of time.
This is a heavy duty lockdown, and it had the desired effect.
It did.
And the mask thing, I mean, masks may help a little bit in certain situations, but in a lot of cases, those same situations, you need distance as well.
That's right.
So it's not one thing. It's not, the magic trick is masks, or, the magic trick is 6 feet of distance. It's the combination. And there's
no magic.
Yeah.
If you don't get it right, then you end up with a lockdown like this, which, that'll do it.
Yeah, you need the masks and the distancing and the de-densifying.
Yeah.
And the hand washing.
The staying outside and not doing things indoors, and all of that put together.
And again, I am deeply sorry to the restaurant owners.
Yeah.
I really.
I totally agree with you.
And we get takeout regularly to try and keep some places in business. There's only so many places you can do that with, though.
Dixon, I got a TV show for you.
Go for it.
It's called My Life is Murder. And it was Ibby and I. There's only one season with 10 episodes. We've watched it. Ibby and I just finished watching it for the second time last night.
Is it on Netflix?
It's filmed in Melbourne.
Oh, great.
Okay. And they actually do such a wonderful job with the production that it essentially features the city.
Nice.
Okay?
Nice.
The city is kind of one of the characters. It stars Lucy Lawless.
All right.
Okay. With another bunch of characters, and it's absolutely, in all ways, absolutely terrific.
Thanks for the recommendation.
Yeah.
I'll do it. Definitely do it.
Need to just remind everyone it's Melbourne.
That's right. Melbourne. It's not Melbourne. It's Florida. Melbourne is Florida. Melbourne is Australia.
By the way, picture.
This show makes me want to go.
This picture of Audrey Hepburn with the face mask was the graphic for TWiV 675. Thank you, Barry.
Yeah, that's
It's a really nice picture. I love it.
Yep.
Dixon, can you take the next one, please?
I can. Emily writes, hello. First of all, thanks for all the work you do. I enjoy listening to TWiV on my runs every week, and I personally think you are the best source of information during the pandemic. Your insights have guided several of our decisions, including keeping our kids home from school and learning online this fall. A few weeks ago, my kids and I were going to visit my parents and in-laws, who we haven't seen for months. So I decided to get a PCR test at our local drive-through state testing facility because I felt it was a responsible thing to do before visiting older family members. I was shocked when I tested positive. While I realized there certainly are theoretical possibilities, we haven't been able to come up with any logical way I could have contracted the virus, as none of us have had any close contact or really any contact with anyone else except each other. We work from home and my kids are doing virtual school. I was completely asymptomatic and no one else in my house had been sick either. I retested negative PCR 48 hours later. I found it interesting that public health had suggested my test could be a false positive. But when I followed up with my healthcare provider, I was told false positives really aren't possible with PCRs. After searching through Google, I found similar media outlets and articles saying the same thing. False negatives are an issue, but a false positive means a positive.
No, a positive means a positive.
Positive means a positive. However, I did run across this recent publication in The Lancet that suggests that false positives are rare, but possible. Okay, and then she gives a reference for that. I understand promoting the narrative of erring on the side of caution. We don't want people assuming they had a false positive and then go out and infect other people if we acknowledge that false positives occur. I also know there are conspiracy theories floating around out there regarding false positives, so I'm curious on your take on this issue. I know there have been several well-publicized events with the NFL where false positives have occurred, both with rapid tests and PCRs. So I find it interesting that there is so much conflicting information out there. I realize that I may never know whether or not my positive was real or not. It doesn't really change anything for me at this point, but it certainly caused a roller coaster of emotions for me, from guilt to frustration to being grateful this ended up being a best case scenario outcome for my family, and if I truly was infected. Thanks again for all you do. Emily from Iowa.
There are false positives for sure. And the way you could figure it out is if you took the PCR product and sequenced it. And
There
nobody
you
does
go.
that because it's too expensive. But so there are a number of ways you can get a false positive. First is error, sample processing error, mix-ups, user error. It happens all the time because there are steps done by humans.
That's
and
right.
last I heard, we're not perfect. Okay? And that's fine.
Really?
That's fine.
True.
We all make mistakes. The second is priming at the wrong site. Priming at a genomic locus can happen under the right conditions. And that's where, if you sequence the product, you could tell. So I had what I think is a false positive. And this is the other thing. If they can tell you the CT value, if it's very high, that emphasizes that it may be a false positive. I would have liked to have my PCR product sequenced, but they don't do that and they don't offer it. So that's why you have to be retested within a day or two, and that resolves it. But there are false positives. It's not correct to say it's not possible to have a false positive with PCR because of what I said.
No, and the simplest scenario is you've got a lab with a whole bunch of samples in it, and you're using an extremely sensitive test, and some of the samples are positive, and of course you can get cross-contamination if things are not done perfectly. But yeah, as Vincent said, there are a bunch of other ways that it can also go wrong. There are procedures that minimize that. So false positives are indeed rare if the test is being done by a good lab, but it's not impossible.
So one of the things I find really annoying about this is that her doctor told her that this couldn't happen.
Yeah.
Right.
Yes.
I can't stand it when doctors are emphatically wrong.
Yes.
Wrong is okay, in particular if they say, I'm not sure.
Right.
But when they're absolutely positive and they're wrong, that bothers me.
Yeah.
And the other thing that occurred to me is that you could follow up with an antibody test. Okay. Now, if you had a mild case, you may not be antibody positive. Okay. But it's another way ultimately to check whether or not you had an infection.
Right. I as you were saying that about the doctor being emphatically wrong, part of me thinks, I'm guessing this doctor has never done a PCR before.
Right.
Themselves in the lab, because I feel like if I asked any of my students, once they'd done PCR a little bit, they would be able to easily tell us that false positives were possible.
Yeah, and the importance of the CT values is if you run a PCR long enough, you can amplify anything from anything.
Well, also, I mean, if you have a high CT value, you could either be at the end of an asymptomatic infection or at the very beginning of something that may or may not be symptomatic. And that's why you need to do another test. And in your case, it was negative, so it was a false positive. But yeah, the antibodies, would tell you if you had an asymptomatic test, and this is the tail end. But doesn't sound like it's. If we had rapid tests that you could do for $1, you could do it yourself.
You just do another one. Yeah.
All right, our last one, Andrew. Hi there, our six-month viewer of TWiV here. I was an auto part analyst in Detroit before the bug. That's a good one, before the bug.
Before the bug.
And I think there could be something I know that may be of good use for you guys.
Wait, did he mean a bug or the Volkswagen? Because he was an auto part, so sorry.
It's an auto part analyst. What do they do?
They analyze auto parts. Sorry, maybe you can write back and say it sounds like an engineering position would maybe be testing them or
maybe, yeah.
like that.
Through my years of work and research, I've begun to realize two things. Car engineers, A, need something that will not break during the 5-year, 100,000-mile warranty period. And B, they want the cheapest option that will fit this zero low warranty requirement. Actually, they could build things that last forever, but they don't want to, right?
Right.
Because
that would not be the cheapest option.
Right.
But also,
I don't think we would want that either.
they could build a car that lasts forever, but then nobody would buy more cars, right?
Right.
Or anyway. Okay, so now on masks. There are 0 washable filters that are deemed acceptable to filter enough fine particulates to protect the engine from damage. There is one well-known filter for being washable. It's called K&N, but this filter needs oil applied properly after washing. Straight from the factory, it still does not filter as well as paper. Long story short, paper works best as a filtration media. And I haven't heard much about filtration quality from anyone anywhere, but I have been way into it actually for years. I actually think the filtration media needs more attention, but what the hell do I know? I'm just an auto parts guy in Detroit. I really don't know what it means, but I thought this was great.
That's great.
Yeah, I think that's great. Andrew, I have been wearing a lot of cloth masks, but I recently got these sort of paper charcoal filters that go into my masks, and they make me feel psychologically like they're doing something. And now you've made me feel like they must be doing something important. So thank you.
You've got a professional opinion that was the right thing to do.
Actually, I understand. He's saying that paper is great. Washable filters are not as good as paper, right? Yeah.
Right. Yeah.
Right.
And he seems to know. He's an auto part analyst, so I think we need to have cross feeding of information,
Absolutely.
right?
Yes. Absolutely.
Yeah, and if you're not working in your auto parts job in Detroit anymore, maybe you look at positions in the mask industry, which is booming these days.
It surely is.
Apparently. All right, time for some picks of the week. Dixon Despomier.
Well, I've been searching the internet routinely for a long time, looking for something that we could all appreciate and laugh at without offending anybody. And I found this particularly amusing. It's a series of photographs taken by some wonderful photographers of animals imitating people doing things that I'm sure will bring a smile to your face, if not an outburst of laughter that's guaranteed to lighten things up a little bit, at least until you can grab that first glass of red wine and sit down and think about the world in another way. So these are real photographs. They're not staged. They're from around the world, different photographers. I just found it very amusing, and I thought I would share that with you.
This was going to be my pick this week, and then I saw you had picked it. Yeah.
Oh, really?
Yeah, I think this overall winner. So these are the 2020 winners of the Comedy Wildlife
Quite
Photography
wonderful,
Awards.
actually.
I think this overall winner, the turtle, ought to be on a poster for ocean conservation.
For plastic.
This is what the ocean thinks of our current behavior.
That's right.
But just a lot of these are really hilarious.
Yeah,
and they're
they're
great
real
photos.
Good. I'm glad.
I also like the smiley fish.
Yes, that's good.
Oh, that was a wonderful one.
I like the video of the giraffe scratching his neck in the tree.
The bear photographs are not so bad either.
Right.
Really
More
good.
subtle, much more subtle.
Brianne, what do you have for us?
I have a talk that I saw yesterday. It was actually live yesterday, and then I was excited to find out that they had recorded it for others to watch. This is the 2020 Christiane Amanpour lecture. about from the University of Rhode Island Journalism School. They featured Ed Yong, who talked about COVID-19 science reporting and a global pandemic. He first talks a bit about the pandemic from his point of view. He does not pull punches. So certainly be aware of that, because there are lots of times where you can tell that, there are some burns going forward. And then he talks about what he has learned about science reporting and science communication, and sort of gives his tips and his things that he's learned. And there are just so many really phenomenal points that he makes that I think many of our listeners would really appreciate. Though, like I said, he does not pull his punches.
He writes for The Atlantic, as I recall.
Yeah. He does. He writes for The Atlantic. He's been one of my favorite
among other
science
places.
Yeah, no, I agree with you.
He's an outstanding science writer.
Totally agree.
He's been one of my favorite science writers for quite a while. I've assigned his book to my students for the past few years. And so I've followed what he's done for quite a while. And I was very impressed with this lecture.
Yeah.
I will listen.
Cool.
This sounds good.
Neat. Alan, what do you have for us?
I have a book that I just read recently. It's called Tree Story. And this, I've been doing a lot of leisure reading lately to just try and do something that doesn't involve any contact with the news. Not sure what got into me that motivated me to do this, but anyway, this is a great read, and it's fascinating science. It's very well written. It's by Valerie Trouet, who is a dendrochronologist. Someone who studies tree rings.
Sure.
And of course, everybody, every 5-year-old knows how you count the age of a tree that's just been cut down. You count the rings. There's a whole science of that. And it's really cool that there's so much more to it. So first of all, they don't go around cutting down trees. They take a drill and take a core out, and then they can count the rings without killing the tree. But they can take samples of trees that fell many years ago and get the rings off of those, and then align them. Oh, well, this series of drought years must be that series of drought years from this live tree. And so they've constructed these chronologies of global weather going back thousands of years. And it's just fascinating. And she tells the story of the science of dendrochronology, and then also a whole bunch of vignettes of history that has been learned through these things, everything from the abandonment of the cliff dwellings in the Southwest and what people lived in them and when, through, the environmental catastrophes that abetted the fall of ancient Rome, and we're getting weather reports from the 1200s or whatever, and she's saying this was a bad year. It's like, wow, you can tell that. So it's a really fun read, and well written, and a great science explainer as well.
Alan, did she cover the story of the guy who determined the age of the oldest tree in the world?
I believe that's in there. Yes. Which one? So he's part of this scene, and he was up on one of the cliffs in Arizona. Yes.
I think the White Mountains of Arizona, they have the bristlecone pines.
Right. And
he tried to take the age of the tree by coring it. And he broke the device, and the device is a $600 instrument. That in those days. And then he had 3 of them with him. He tried all 3 and they all broke. So he ended up cutting the tree down.
Yeah, there's the story is in there. He cut the tree down. He got a permit to do so. He cut the tree down. This was in the early days of dendrochronology.
Exactly.
And then back in his hotel room that night, because this was a field study, he was counting the rings, and he realized to his horror that he had just cut down the oldest known tree in the world.
Exactly.
And now they've subsequently found older trees in that same area, but he then left the field of dendrochronology and has gone on to other areas of science, and really doesn't want to talk about that particular.
No, he does not. But he's changed his name also.
Yeah, there are a lot of other very interesting stories in it.
Yeah, I'm going to buy that book. That's a great book.
I didn't realize that dendrochronology was the study of rings, and dendritic actually means having a branched form resembling a tree.
Resembling a tree.
And there you go for dendritic cells. That's
Indeed.
funny. Rich, what do you have for us?
I have a website, a thing called Violin, the Vaccine Investigation and Online Information Network, that I ran across. As I said earlier in the show, I was sort of buffing up my knowledge of vaccines and in particular viral vectored vaccines, and I ran across this thing. It's curated by a group at the University of Michigan. It's been out there since 2008. And it looks like I even communicated with the head guy to make sure that this was not just something that was lying dormant. And no, they're still actively curating this thing, and it seems up to date. Relative. There's a couple of holes in it that I discovered, but it's pretty comprehensive. Cool. And I was for my purposes, I was particularly interested in a subdomain called VaxVec. Let me see if I can find that here. Yeah, VaxVec, which is a database of recombinant vaccine vectors and recombinant vectored vaccines. Nice. So, and this whole thing is searchable as well. They have a section on what's the status of the COVID vaccines and et cetera. So I think this is a potentially useful resource.
This is deep.
Cool.
I just started clicking around randomly, and I'm instantly way into an extensive description of dengue virus biology. It's wow.
One of the nicest things is that it's searchable. Okay. So you can try and tease out of it something that you're particularly interested in.
And you can specify the subsection of the site that you want to search, because that is apparently an issue here. This thing is huge.
And it also lets you sort of do things like blast searches against vaccine immunogens and things like that. That's really cool.
Wow.
It's neat. Very neat.
Cool.
My pick is a website called Wearing is Caring. This is put together by a team of scientists here at Columbia University. They've created a set of mask wearing guides. They have, myths busted. They have misconceptions like, how should I wear a mask? How do I wear it properly? Should kids wear a mask? Why do public health guidelines change? Anyway, good resource.
Excellent.
From my colleagues here at Columbia.
Cool.
Nice.
They're also on Twitter, Wearing is Caring, and other social media. Check them out. All right, now, as many of you know, I'm going to give away some books, some Principles of Virology edition number 5. Authors get books. I don't need all of them, just need a couple copies. So I'm going to run a bunch of contests, and the first one is to write a pandemic poem. I think it's clear what that means. Any form. Could be a limerick, it could be haiku, regular, iambic pentameter, whatever you want.
It has to be a poem that spreads from one person to the next and then goes around the world.
That's
and
right.
you cannot stop it.
Yeah, viral.
Exactly.
All right. So
it has to go as fast as a bat out of hell.
So we're looking forward to reading these. We'll read them, and amongst us, we'll decide which one we like. And we've done this before, and we got some wonderful submissions. So send the poem. You got to follow the rules now, okay? Otherwise, we won't consider them. All right, I will consider them. All right. TWiV@microbe.tv. So send them to TWiV@microbe.tv. You have to send them to that email. And you have to have the subject line poem. Otherwise, I won't see them amongst all the other stuff. Okay?
Right.
TWiV@microbe.tv poem. Really simple. And anybody could enter. Okay? TWiV is at microbe.tv/TWiV, which you'll find all the links about all the stories that we talk about, and also the emails and stuff. And if you want to send us questions, comments, TWiV@microbe.tv. But be nice, okay? If you have a disagreement, that's fine, but just be nice. And if you like what we do, well, for the month of November, go over to parasiteswithoutborders.com and donate, and they will match the donation. But if you don't want to do that, that's fine. You can go to microbe.tv/contribute and donate to us. That's cool as well. Dixon de Palmier is right across the river here from me. And,
I am.
And he's also...
And the sun just came out. The sun just came out.
I see. Look at that. Finally, the stinking rain for 2 stinking days, knocking all the leaves off trees. Just a mess. Now it's nice.
And you've got a backyard to clean up when you get home.
You know, one year I bought a snowblower and it didn't snow all winter.
Of course.
So you blew leaves.
So this year I bought a leaf blower for the first time, but the bloody leaves are still falling. I don't get it.
Well, I thought you were going to say then it snowed.
You thought the leaf blower was going to keep the leaves on the trees?
Well, this is scientific logic. If a snow blower stops snow,
I guess so.
a leaf blower.
A leaf blower should stop the leaves.
This is all true. This is all true.
I just mow my leaves.
In Texas, you don't need leaf blowers, right?
Not true. The leaves do fall off the trees.
Oh, yeah. You have live oaks and stuff.
They don't have this as much of this sort of last colorful gasp. There's some of it that goes on. That's right. You told
me that. But by the way, I take my leaves and I blow them into a pile, then I chop it up and put it in a compost pile.
Oh, good.
Wow.
And then I have dirt. This year I built 3 big compost things. I took down a wood cedar fence. You know, cedar lasts outside, and I made the sides of 3 compost bins and I love making dirt. It's so cool.
I just mulch them in situ with the mower.
You can do that too.
I just keep mowing my lawn every couple of weeks until Thanksgiving or so, and they go away.
Yeah.
So Vincent, next spring you should hang a shingle out in front of your house that says "live bait sold here," because you'll have tons of worms in those compost heaps, and the fishermen will just flock to your house.
Yes, you do want to have worms, actually. Yes. Anyway, Dixon, besides being across the river, he's at Racaniello.org and thelivingriver.com. Thank you, Dixon.
You're welcome. This is great.
Brianne Barker's over at Drew University. On Twitter, bio Prof. Barker. Thanks, Brianne.
Thanks. It was great to be here. And I'm kind of sad because we don't have the sun here.
Keep waiting. We'll bring it to you.
I'm sure it'll come.
I'm surprised your face doesn't light up the entire portion of the place where you live. You have such a wonderful smile. I must say that.
Thank you.
You do.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's over at alandove.com, and on Twitter, he is Alan Dove. Thanks, Alan.
Thanks. It's always a pleasure.
And now we have everyone except Brianne with the high-definition camera, and I will get you one this week.
Awesome.
And we will all be crystal clear. At least in our pictures, not always in our speaking.
Not always in our explanation.
That's right.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.